Ladies and gentlemen, good morning and Assalamu alaikum. Welcome to the first session of the fourth day of the STEM Education Training Program funded by British Council. Let's start the session with the name of Allah Almighty.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الله الذي خلق السماوات والأرض وأنزل من السماء ماء فأخرج به من الثمرات رزقا لكم وسخر لكم الفلك لتجري في البحر بأمره وسخر لكم الأنهار صدق الله العلي العظيم.
Jazakallah. Ladies and gentlemen, today we have with us Dr. Noreen Latif. She is Assistant Professor at Center of Excellence in Molecular Biology, University of the Punjab. She did her PhD from the same institute and joined as postdoc fellow and later as assistant professor. Her area of research is regenerative medicine for the repair of damaged organs. She has produced 19 MPhil and 7 PhD scholars. Today, she will deliver her talk on stem cells research bench to clinics. Welcome, ma'am. On the behalf of Virtual University of Pakistan, please proceed for your presentation.
I'm Dr. Noreen Latif. Yes. Okay. My name is Dr. Noreen Latif. I am from CEMB, Center of Excellence in Molecular Biology, University of the Punjab. It is a state-of-art institute in the field of molecular biology. And we are producing MPhil and PhD scholars, and we are proud that our graduate students are serving around the country as well as around the world in the prestigious institutes. And one of my students, Dr. Azam, is here, and he invited me to give a talk on urgent basis. And yesterday, he asked me to give a lecture. So in urgency, I prepared something for you. And my today's lecture is about stem cells research from bench to clinics. The topic I selected is based on my area of research. My area of research is stem cells biology. We in Center of Excellence in Molecular Biology are the pioneers of the stem cells research in Pakistan. And we started in way back in almost 2000 or 2001. And now different other departments and other universities as well are doing research on the stem cells, as well as different clinics are importing stem cells, and they are telling their patients that they are importing stem cells from different international companies, and they are transplanting the stem cells in the patients for the treatment of the different diseases like osteoarthritis, spinal cord injury, infertility, baldness, and skin regeneration, as well as for burn patient, etc., etc. So I will not comment on that, whether they are truly transplanting the stem cells in the patients or not, and they are taking a lot of money from the patient as well. But we are working on the grounds, and we have published many international publications in international journals with good impact factors, as well as we are in collaboration with different hospitals in Pakistan and around the world, and we are moving from bench to the clinics. And currently we are in collaboration with Lahore General Hospital, Lahore Jinnah Hospital, LRBG Hospital, Ghurki Hospital, etc. This was the brief introduction of mine. So I come to the point that what are stem cells and what are their importance, and how a little bit about the research that we are doing there, as well as what are the clinical aspects of the stem cells and what people around the world are transplanting or implanting the stem cells for the treatment of different chronic as well as acute diseases. So first of all, maybe you all know or might know, don't know that what are stem cells. Stem cells are the cells. I was smiling because many familiar faces are here. I know Azam, Sumera, Samina, and one of person sitting behind, I don't know his name. I know many people now. So stem cells are the specialized kind of cells. They can, अगर मैं यहाँ आ जाऊँ तो मेरी आवाज ऊंची आ जाएगी आपको, तो इशू तो नहीं है, रिकॉर्डिंग में कोई इशू ना हो तो मैं इधर आ सकती हूँ. अच्छा, okay. Stem cells are special kind of cells. Okay, that has unique capacity to divide and produce cells like themselves and specialized type of cells also. So stem cells. हाँ जी, अब आ रही है आवाज. Stem cells are special kind of cells that can renew themselves as well as they can produce other kind of cells, means they can produce cells which can be differentiated into other kind of cells, or they are specialized cells. As they are uncommitted cells, they remain uncommitted in our body until and unless they get some kind of signal and they get differentiated into the specialized kind of cells, like they can be differentiated into heart cells, into the liver cells, into the cartilage cells, into the eye cells. So they are uncommitted, but they can get specialized or differentiated when they get some kind of differentiation signal, and they can be differentiated into specialized tissue and then later on into the organ. So stem cells are basically a revolutionary idea that it is now prevailing into the medicine, that in medicine we try to regenerate the organs or replace the organs. But the idea behind the stem cells regeneration potency or regeneration science is that we try to repair the organ by itself or regenerate the organ by itself, so we do not get the need to transplant the organ. For example, if the liver is damaged inside the body, there is a natural phenomena of regeneration, but if that phenomena is not properly working, and we are taking the complementary medicines as well, but we need something else that we do not want to replace that liver, then we can transplant the stem cells. We can isolate the special kind of stem cells from the body, and then we can either differentiate those stem cells into the hepatocytes, which are the cells of the liver, and or either we can even transplant simple stem cells into that liver, and then the liver stem cells will divide there. They produce some special kind of paracrine effect, means they produce some special signals which help the transplanted cells as well as the cells of the liver to get mature, to get divided, and so they can produce some factors, and the regeneration process starts, and the organ is not needed further to be replaced. Stem cells has the properties that they are self-replicating, they are unspecialized cells, they are capable of generating specialized daughter cells or progeny, and they are plentiful in embryonic development, but in the later stages, they are less in numbers, and in the children, they are more active, but with the passage of the time, as we grow older, then they are less effective and less active, you can say that. So in that case, we need the isolation of the stem cells from the same body or from the any other organ or organism or the human being, and then we can grow in the lab, and then we can transplant them back into that human. They have the capacity for self-renewal, and but that self-renewal capacity decreases with the time or aging. They can be categorized as embryonic or adult stem cells, and I will discuss it in the later slides. They have possess specific markers by which we can recognize them, we can characterize them. So whether they are true stem cells or they are some other kind of mature cells or not. They exhibit varying plasticity depending upon the environmental and commitment level. When the stem cells, there are two kind of division. One is called as the symmetrical division and other one is called as the asymmetrical division. In the symmetrical division, when the one cell divides, it produces the same kind of cells later on, and the daughter cells will divide and it will produce the same cells later on also. But in the asymmetric division, when stem cell, because the stem cells has the asymmetric kind of cell division, when the stem cells divide, it produces a cells like the parent cell, which is called as the stem cells, and a other kind of cell, which is called as the progenitor cells. That progenitor cells has the ability so that it can be differentiated when it gets some kind of signal or some kind of markers or some kind of information that you need to be differentiated, then that progenitor cell will be differentiated. You can see that the blue color is for the stem cells and the red color is for the progenitor cell. So the stem cells has the ability of asymmetric, is divided by the asymmetric division, and it produces the stem cells like itself as well as the other cell, which is called as the progenitor cell. What are the progenitor cells? You can see that stem cells divide and produces the stem cells and one other cells, which is called a specialized cells, but the progenitor cell, which is some kind of specialized cell, it will only divide to produce the specialized cells, so that it cannot produce any stem cells. So where the stem cells are present or where the stem cells reside? You all know that the life starts with the fusion of the sperm and egg. When the egg divides, after the fertilization, when the zygote divides, then first there is a one cell stage, then 2 cell, 4 cell stage, and so on. So when there is a zygote, then which is the fusion of the sperm and egg, then it has the cells which, when it is at the stage of 5 to 7 days of embryo, you can say that the 5 to 7 day of zygote, then it has all type of original cells that can produce the all human beings or all any other organism later on. So the cells present at the 5 to 7 day of a zygote or an embryo, that is called as the embryonic stem cells. These cells are totipotent and they can produce the whole organism. But after that, you can say that 5 to 6 day of 7 days week of fertilization, there are cells which can divide and produce different organs. They are called as the embryonic germ cells, and they are pluripotent, and they cannot produce the whole organism, but they have the potency to differentiate into many organs, and it can help in the regeneration of the many organs later on also. And the pluripotent cells, they are present inside the embryonic germ layers, and they have the 3 germ layers, which are the mesoderm, endoderm, and ectoderm structures layers. And you know that these 3 layers are important part of all the organisms. So what are the difference between the totipotent, pluripotent, and multipotent stem cells? Totipotent are the cells, they are capable of any fate, means they can generate all cell type necessary for the development of any organism, which I told that they are present inside the, we can isolate these embryonic stem cells which are totipotent at the 5 to 7 day of the fertilized embryo or zygote. Well, the pluripotent stem cells, they are cells or stem cells which have the possibility to produce any other tissues and organs, but they have some restrictions and limitations that they can produce many kind of cells or organs or tissues, but they cannot produce the whole organism. Multipotent, ये आपका थोड़ा सा खराब हो जाता है, बीच में चलता नहीं. Multipotent, they have limited to low number of specialized cell types in differentiated tissues. Like we can isolate the stem cells from our cells also. We have stem cells in our eye, we have stem cells in our teeth, we have stem cells in our hair follicles, even our fat in the umbilical cord, which is get wasted after the child delivery, in the umbilical cord blood, as well as from the liver tissue, from the any other tissues which are present from the heart as well. But all these organs have stem cells, but they are limited in number because they are developed organs. They have limited number of stem cells, but when there is an injury or any disease, then these stem cells get some signals from our body, because there is a process of homeostasis that body try to heal itself by first, and then when there is not healing and by the body, then we need to take some medicines to heal our body. So the stem cells are present in the mature organs also, but they are limited in numbers, and we have to isolate them by passing the special kind of procedures in the lab, and then we have to characterize the cell, whether they are stem cells or they are the part of the mature cells or mature organ. So they are multipotent. For example, if I want to isolate the stem cells from our fat, which I will discuss it later, we have abundance of fat, like me, including me. So we are very, God is very helpful, and He has provided stem cells, even the fat also. So we can isolate the stem cells from the fat, and then we can characterize it by different markers which are present on the stem cells, and then either we can transplant those stem cells back into my body in the disease organ, or we can differentiate those cultured stem cells in the lab, and then we can specific kind of tissues or specific kind of cells, differentiated cells, and then we can transplant them back into the disease organ. And then when we transplant the cells in the disease organs, then there is a paracrine effect. What is paracrine effect? That it recruits the different kinds of growth hormones and different kinds of proteins, different kinds of products toward that transplanted organ or disease organs where we have transplanted the stem cells. Then those signals, those proteins, those growth factors will help in the regeneration of that organ. And the stem cells, they are present there, they can also get differentiated there into the specific organ, and then they can start the process of regeneration. But in the adult cells, they have limited potency to get differentiated. So we can call them, they are multipotent stem cells. This is the image, very good image that shows that what are the sources of the stem cells and what are their attributes or the potency. You can see that these are the stages of the human development. This is the single cell embryo, then 3-day embryo or 5 to 7-day embryo. When this stage is achieved, which is 5 to 7 days, then we can isolate the cells from this stage also, and these cells will be called as the embryonic stem cells. They are totipotent. Why? Because they are the origin of the development of the whole human being. So they have the potency to get differentiated into any kind of cells. So there is a debate on the use of these stem cells. Being a Muslim, we also have some ethical issues, as well as the European or international societies. They also debate on the use of these embryonic stem cells. So there is no permission around the world in most of the countries to use these embryonic stem cells. So mostly people are working on these fetal stem cells and adult stem cells. We in the camp are also working on the fetal stem cells or the adult stem cells. Mostly they are called as the adult stem cells, and we are not working on the embryonic stem cells because in our religion we believe that if we destroy this embryo, that it will be the killing of a human being, which is not permissible in our religion. So we do not go for this. As you know, how the people can isolate the cells from these embryos. As you all know, there is a procedure of IVF transplantation, which is called as the in vitro fertilization. When there is a fertility issue, then there is a facility available to that we can go for the IVF also. In the IVF procedure, there are many numbers of the embryos of the eggs and the sperms, and some of them are used later on and the other get wasted. And the doctor will see the healthy embryos or fertilized egg and transplant into the ovary of the patient, and the other embryos or other fertilized egg, they get wasted. So the people think that they are getting wasted, so we can isolate the stem cells from them, and then we can use it for the research purposes and for the benefit of the human being to cure different diseases, etc., etc. But we are not working on it. And the fetal tissue and the other tissue which or cells which are called as the embryonic germ cells, and they are pluripotent in nature. We can isolate it from the 6 weeks embryo, and we are not working on those tissues or cells. And the fetal tissue, which are pluripotent or multipotent, they are called fetal because they are isolated after the fetal delivery. Like when there is baby delivery, then there is a placenta and the umbilical cord by which the baby is attached to the mother womb. Then they get wasted. But that umbilical cord or the blood which is present in the umbilical cord, which is called as the cord blood, or even the placenta around the baby, they are very good source of stem cells because they are close to the embryonic thing, but they are not the embryonic because they, as the baby is born, it has a big capacity to develop it into a human being and big regeneration potential also. So the tissues or the samples which we take from the hospital, as I told you that we are in collaboration with Jinnah Hospital, LGH Hospital, Gangaram Hospital, then what is the purpose? First purpose is to take the umbilical cord from them. We take the umbilical cord, which is get wasted after the baby delivery. There is no use of it. We take that umbilical cord and then we use it for the isolation of the stem cells. And the other thing, we are in collaboration with the different private clinic, and there is a procedure which is called as the liposuction to get away from the fat from your body. So it is also a waste product. We take that lipoaspirate and then we isolate the stem cells from that lipoaspirate. They are lipoaspirate is some kind of matured or adult tissue, while the fetal tissue, which is the umbilical cord, is more close to the embryo, but it is not embryo, but it has a potency. It is multipotent or some kind of pluripotent, and it produces the cells in a lot of number, and it can be transplanted to cure different diseases. And in the adult, you can see that we can isolate the stem cells from the cord stem cells or placenta stem cells. And in the human being, we can isolate the stem cells from the any other portion, like bone marrow was the first source for which the stem cells are isolated, but it is a difficult procedure. It is a more intensive procedure. So we do not go for that bone marrow isolation nowadays. We have switched towards the lipoaspirate and the umbilical cord now. This slide shows what is the difference between the embryonic stem cells, embryonic germ cells, and adult stem cells. The main difference is the source from where we are isolating it. That is inner cell mass, the primordial germ cells or gonadal ridge of the 5 to 10 weeks fetal, and it is the adult organs. What is the adult organ? Bone marrow, blood, cornea, retina of the eye, brain, skeletal muscle, dental pulp, liver, skin, lining of the gastrointestinal tract, and pancreas. You can see that there are a lot of organs in our body where the stem cells are present, and they can produce stem cells later on in our if we have some disease, and we can go in lab and transplant it back into that organ, into the human being. And it has the potency unlimited production of the cell, that it has unlimited number of symmetrical divisions without differentiation. It can be divided into for the few years. It will produce the cell like itself, so it will not get differentiated. The growing cells will not get differentiated because they are more to the like, and they are pluripotent. And the other thing is moderate number of symmetrical division without differentiation, 7 to 80 doubling population है. They have 22 years of doubling time and they have 70 to 80 doubling time के आप इससे ज़्यादा अगर इसे करेंगे तो फिर ये maturation की स्टेज पर जाना शुरू हो जाते हैं. जबकि इसमें आप जो है लिमिटेड नंबर ऑफ डिवीजन है, आप कुछ साइकिल्स जाते हैं. लाइक हम जब सेल्स को ग्रो करते हैं हम अपनी लैब में, तो उसको मुख्तलिफ पैसाजेस, मतलब हम जब ग्रो कर जाता है एक खास नंबर पे हम उसको दोबारा से डिटैच करते हैं, फिर अगली स्टेज पे ले जाते हैं, उसको हम पैसाजिंग कहते हैं ट्रांसप्लांटेशन के बाद. तो हम आमतौर पर जो यूज करते हैं वो हम P2 P3 के सेल्स यूज करते हैं, बिकॉज वो उसमें से जो आपके मिक्सचर ऑफ सेल्स हैं, जो अनवांटेड सेल्स हैं, जो प्योर नहीं है, वो निकल जाते हैं और अच्छा प्रोलिफरेशन पोटेंशियल उसका आ गया होता है. उससे आगे भी हम 4 तक भी जा सकते हैं, लेकिन कोशिश करते हैं कि वहां रहें. आगे आगे जैसे जैसे हम जाते हैं तो उनमें मेच्योरिटी आती जाती है और उनका जो है वो कैलिंग पोटेंसी कम होती जाती है, उनकी डबलिंग टाइम भी बहुत बढ़ जाता है और उनका नंबर ऑफ सेल्स कम होता जाता है. तो इसलिए ये जो, क्योंकि एडल्ट सेल्स हैं इनकी कम पोटेंसी है, इसलिए इनका जो टाइम जो है लिमिटेड है और उनमें कम प्लास्टिसिटी पाई जाती है. इस तरह से बट एम्ब्रियोनिक स्टेम सेल्स का क्योंकि अनलिमिटेड ग्रोथ है, तो इसका एक जो मेजर ड्रॉबैक है दैट because you know that carcinoma or cancer is also unlimited growth of any tissue or any cell present in our body. So there is a chance that कि आप जब उसे ट्रांसप्लांट करें तो वो अनलिमिटेड ग्रोथ हो जाए सेल्स की, और वो वहां पर जो है वो बहुत सारे नंबर जो है और वहां पे कैंसर डेवलपमेंट हो जाए. तो ये इसकी एक मेन जो है वो उसकी रीजन भी है कि इसको नहीं यूज किया जाता. यहाँ पर नहीं है और यहाँ पर भी नहीं है. और जो OCT4 है और जो दूसरे फैक्टर्स हैं, ट्रांसक्रिप्टिव फैक्टर्स, ये सब उसमें मौजूद हैं. इनमें भी स्पेसिफिक मार्कर्स हैं, इसके एम्ब्रियोनिक के और मार्कर्स हैं, मैच्योर के और मार्कर्स हैं और फीटल की साइड के और मार्कर्स हैं. मार्कर्स क्या है कि हर चीज को आपने रिकग्नाइज करना है. मुझे रिकग्नाइज करना है मेरी एक खास शक्ल सूरत है, मेरी एक खास रिकग्निशन है कि नाक ऐसा है, ये तिल का निशान है, ऐसे ऐसे. लेकिन जब ऐसे आप सेल्स को रिकग्नाइज करते हैं, आपने फैट में से सेल्स लिए हैं, फैट में एडिपोसाइट भी हैं जो कि आपके फैट के टिश्यू हैं. सब सेल्स जो आप आइसोलेट कर रहे हैं, दे आर नॉट स्टेम सेल्स. तो स्टेम सेल्स के स्पेसिफिक मार्कर्स हैं ऑन दिस सरफेस व्हिच आर कॉल्ड एज द मीसन, मे बी कॉल्ड एज द मीसेनकाइमल स्टेम सेल्स मार्कर. CD90 है, 105 है, 73 है, और कुछ ऐसी है. फिर जब पॉजिटिव को चेक करते हैं तो नेगेटिव भी चेक करते हैं कि उनमें ये वाले मार्कर्स तो नहीं है. लाइक ज्यादातर हम हेमेटोपोटिक मार्कर जो ब्लड के मार्कर्स होते हैं वो चेक करते हैं कि वो तो मार्कर्स नहीं हैं, प्योर सेल्स हैं, और फिर हम आगे उसको जो है वो लेके चलते हैं. This is a cartoon just to summarize the what are stem cells. That if the stem cells grouping center asks the question that can you divide and renew yourself, he says yes, but he says that I am still here. But after that, you can see that I am unspecialized, but my brother, which is like me, it can be become specialized, and it can be differentiated when some get some signal or some proteins or some information to get differentiated. So my brother or the daughter cells, like the original stem cells, it can become a skin cell, a liver cell, or any other kind of cell. And he specialized to form tight junctions to his neighbors. You know that cells have some junctions or tight junctions, and they can attach themselves with each other. So this is just a nice cartoon to show you that what are the stem cells. They are unspecialized. They produce some kind of cells also, cells like themselves as well as other cells which are called as progenitor cells, and that they can be differentiated. That progenitor cells get differentiated into specific organs. What is human embryonic stem cells? You can see this is the summary of the previous slide that with the IVF treatment, donated IVF embryo, which is not longer required, we can isolate that stem cells or embryonic stem cells after 5 to 7 days at embryonic stage, and then we can culture that cells into in our laboratory, and then we can use that cells from different purposes like research, normal development and understanding the disease, procedure, molecular biology of the disease, and developing therapies to treat different diseases, as well as we can use that for the discovery of different type of drugs also. And other things that we can do with these embryonic stem cells, that, for example, if there is any kind of gene which is mutated and which is the main reason for the cause of different diseases in that or human being, and we do not want to that disease to get transferred into the next progeny, that we will want to replace that gene. In that case, we can do the gene therapy or do another procedure which is called as the somatic cell nuclear transfer. In that procedure, we take the donor cell as well as the any other egg, and then we we remove that disease nucleus in which the DNA is present, the mutated DNA is present, mutated gene is present. Then we replace that nucleus with the nucleus from any healthy donor, and which is called as the somatic cell. Somatic cell means we have taken the nucleus from a somatic tissue, like skin tissue or any other tissue, and we have replaced that nucleus from the somatic tissue into the egg, and then it will divide, and then the morula or blastula stages will go on, and there will be development of a human being or fetus, which will not carry that specific disease which is present inside the egg of that of the mother. The other tissue kind of cells which I told you that are called as the tissue specific cells, they are also called as the adult cells, but mostly we call them adult cells, but they may be they are more likely to be tissue specific cells, because we isolate from the adipose tissue, we isolate from the liver tissue, we isolate from the cartilage tissue, from the heart tissue, from the eye, etc., etc. So they are called as the tissue specific cells. They involve in tissue hemostasis and repair. They are generally multipotent, and they because I told you that they are less in number in the organs, so they are more difficult to there is more difficulty in isolation of the stem cells from the tissue or adult tissue. But we are very expert in that. Induced pluripotent stem cells. As you know that adult tissue are unipotent or bipotent, or some you can say that multipotent, but they are not pluripotent. They cannot get differentiated into multiple organs or many organs. There is another kind of stem cells which are manipulated to be like pluripotent. These tissues or these cells are called as the induced pluripotent stem cells. As the name depicts that they are induced to get pluripotent. Means we can take the cells from the patient, like mostly the first experiment was on the human skin fibroblast cells, and there was a Nobel Prize for this procedure, which was IPSC. Yamanaka from the Japan was nominated and get the Nobel Prize for this research for the production of the induced pluripotent stem cells. For example, we take a skin tissue from the patient, and we take the fibroblast from that skin tissue, and we grow that skin biopsy in the culture dishes in our lab, and then we provide that fibroblast tissue with some transcription factors. These 4 transcription factors are very much important: CMEX, OCT4, KL4, and SOX2. The combination, excuse me, the combination of these 4 transcription factors causes the reversal of these matured fibroblast cells toward the pluripotency stage. And then these pluripotent stem cells, which are patient specific, we have taken it from the patient, and then we can do multiple of things with that induced pluripotent stem cells. We can either use gene targeting to repair the disease gene in that patient, or either we can differentiate that pluripotent stem cells with the help of some signals, some compounds, and some other materials, so that we can use some specific drugs, and then we can transplant that back into the patient. So you can see that we have used the gene targeting and repaired the iPSC cells, and we have then differentiated into the skin cells, and then have transplanted back into the patient. So means we can take the skin cell if there is a disease of the skin, and it is not possible to cure the disease with the help of that some kind of drug, etc., because that is the cause is the gene mutation, and gene is disease. So what we do, we can take the diseased skin from the patient, and then we can revert it back into the pluripotent stage, and then we can repair that gene, and then we can differentiate it back and then transplant it back into the patient. And the other thing that we can culture in, and then we can use the different compounds to differentiate it back into the skin cells, and then we can use different kinds of drugs and therapeutic compounds, and then we can treat it back, that tissue back into the diseased human beings. So what was the features? They can create stem cells directly from a patient, and they can be maintained indefinitely in the laboratory, and then pluripotent with similar properties to embryonic stem cells. So if we cannot use the embryonic stem cells, but we have one solution to that problem, that we can revert it back into the pluripotent state, and then we can put it back into the patient. So what are the different attributes of the embryonic stem cells, iPSCs, and tissue-specific stem cells? The source is, as you all know now, and the rate of proliferation is very high in these cells, and it is very low in the adult stem cells. The availability is high, but it is low here, and spontaneous differentiation is high, and it is very low in tissue-specific or adult stem cells, and they can produce diverse cell types, and high in the iPSCs and ESCs, and low in the adult stem cells. So you can see that all these properties are very similar to each other. So we now we do not need embryonic stem cells, so that we can transplant it back into the patient, and we can regenerate the tissue of the organ of a specific patient. So why the stem cells are important? Because they are present in the muscle, neuronal, and endothelial hematopoietic cells, and we can use any kind of stem cells from different purposes. One is good, one is something somehow not good that much, but there is no specification or no limitation that we use that only kind of stem cells, and we can use only pluripotent stem cells, and if there are no pluripotent stem cells, then we cannot transplant into the patient, and there is no regeneration process. In the last slides, I will show you that people around the world are using normal adult stem cells, lipoaspirate, bone marrow stem cells, and other kind of stem cells, umbilical cord stem cells, for the regeneration of different chronic diseases in the patient around the world, and they are getting benefit from that transplantation also. So new drug can be discovered with the help of these stem cells. We can use it in the genetics, molecular biology, biological control of tissue growth, in vitro and vivo systems, and cell therapy. I told you that in previous slide, there's stem cell nuclear transplantation or cell therapy, gene therapy. they can also be done with the help of the stem cells. So there are 3 to 4 slides. I want to give you some introduction, what the research at the CAM currently we are doing on. This is the building, very beautiful building of our prestigious institute, CAM. As I told you that we do not use embryonic stem cells. We use only adult stem cells. In start, we started from the bone marrow and bone marrow of the rats. We have a very good state-of-the-art animal house there. We have rats there. We have mouse there. And we, as I told you, that we started the research there around 2000 and 2001. And at that time, there was no know-how of the what are the stem cells. So that CEMB is the pioneer of the stem cells research. In start, we started with the bone marrow of the rat and mice, and we isolated the bone marrow stem cells from that, because around the world, the main source of the stem cells isolation at that time was the bone marrow. But it was a very invasive procedure, and we also took one sample from the Dr. Amir. He is very non-orthopedic and spine surgeon, and but the procedure is very invasive, and so we do not go for that, and later we switch to the other tissues also. So the other issue is nowadays we are working on the basically umbilical cord as well as on the lipoaspirate. So you can see it that this is the umbilical cord, the cord by which the baby is attached to the mother womb, and it is the main route of the infusion of the different nutrients and the excretion of to the mother. And with the help of this umbilical cord, baby get all the nutrients, and all the waste materials are excreted out with the help of this umbilical cord. We take that umbilical cord from the patient's C-section, basically C-section deliveries, because in the normal delivery there is a chances of contamination. So we avoid that, and then we take that umbilical cord in a very sterile PBS solution, and then it is taken back to the laboratory. Then after different washing and other things, we chop it. Dr. Sana is sitting here, and she has worked a lot on this umbilical cord. Or you can see that we chop it with the help of the scalpel, with the help of the knife, and there is a vein inside that umbilical cord, and we remove that vein, and after chopping and mincing, and we add some also enzyme in it, and get the collagen inside that umbilical cord is get digested, and after that we strain it, and then we spin it, and there is a pellet, and then we grow that cells in our special medium, and with some kind of growth factors and FPS, and then after some days, within almost 1 month, there is enough, you can say, confluency that we can use it for the even transplantation procedure in the human beings also. And then you can see that. So please, देख लें, ये नहीं चल रहा, कभी चलता है कभी नहीं चलता. Or this is the culture fast, and after that, after almost 15 to 20 days, there is a colony formation of the tissue of the cells, and then we do different characterization procedure to make it confirm that the isolated stem cells are the umbilical cord stem cells. The cells isolated from these umbilical cord are called as the mesenchymal stem cells, and we use different mesenchymal stem cells marker to make it confirm that these are the mesenchymal stem cells. The other source is lipoaspirate. There is a very funny picture that, as I told you, that we have abundance of fat in our cells because of our lifestyle, because of the very less active lifestyle. Also, we get seated on the chairs, and then we have a laptop in front of us, and we have burgers and etc. All that. Okay, so there is a lot of source of stem cells in our cells also. So this pictures only shows the fat tissue, but now we are not using the fat tissue, but we are using the picture on the top of the right side is the lipoaspirate. We wash it, we take it from the hospital, and then we extensive washing and other things and other, there is a long procedure, and then we take the pellet and then we culture the pellet in the culture flask, and then we grow the lipoaspirate, which is called as the adipose derived mesenchyma stem cells in our lab. You can see these are the picture of very nice picture of the mesenchymal stem cells. They are fibroblast or spindle shaped in structure, and we get very happy to see these cells coming out in our culture class. Then we, as I told you, we use different stem cells markers. We use immunostaining procedure. We use antibodies, and then we stain with the fluorescent dyes, and then we check it in the under fluorescent microscope and check the there is a fluorescence of the dyed or immunostained cells, or there is a procedure which is also called as the FACS analysis, which by which we sort the cells that whether they are dyed to the specific antibodies which are mesenchyma specific, like CD4090 positive, 90, 73, etc. But they are also negative markers like CD34, etc., etc. And there is also another procedure, that is, I told you that 3 mesenchymal stem cells has the nature to get differentiated into the 3 germ layers: epidermis, endodermis, and mesodermis. So we can also differentiate these original MSCs into 3 lineages, so that we are dead sure that the cells that we are working on are the mesenchymal stem cells, and there is no impurity in that cells. This slide is just a brief outline of the research that is being done by me and by my group. So we are basically working on 3 organs. One is the cartilage, and other is the spinal cord, and third one is the cornea. So the main theme is we take the samples from the human tissue, umbilical cord, and lipoaspirate, characterize it, and then we use different modalities or different procedures. We use different compounds, and so that we can either improve the potency of the stem cells also. So what is the logic behind them? For example, we are planning to transplant the cells into any human beings or even in the rat model of any disease. First of all, the first procedure is to take the trypsinize the cells, pellet it down, and then count the cells, and then inject the cells into the specific disease organ. But you know that when we do the injection, there is many chances that many of the transplanted cells get wasted with the all the circulatory system, with the injection, and etc., etc. They may not reside there for a longer period of time, so that they can do their function properly. So for that purpose, initially we were transplanting the stem cells into the any disease organ. Still we are transplanting it, but there is also a need that there should be some kind of scaffold, there should be some kind of bedding that we can grow the stem cells on it, and then we can transplant it back into that disease organ. This is the reason of using different kinds of scaffold and for the purpose of transplantation. So me and my other colleague, we are working on the stem cells, mesenchymal stem cells, as well as on the scaffold also. And we are working on spinal cord, osteoarthritis, cartilage disease, for the skin burn patient, as well as from the for the liver regeneration procedure or liver cirrhosis, as well as for the diabetic foot ulcers also. Diabetes, there is, you know that there is a disease in which diabetes, there is ulcer formation mostly on the patient's foot, does not get cured, and there is a very problem to the patient, and in some time patient has to cut his knee or foot So to get rid of that disease part, and which is a very painful procedure. So we try to help the patient for the treatment of that diabetic foot, as well as for the burn patient, as well as for the osteoarthritis, as well as for the damaged spinal cord injury, etc. So we are working on scaffold, we are working on different natural compounds as well as the biopharmaceutical compounds to increase the potency of the stem cells, as well as we are working on the phytoextract. And I would like to share that recently I got an RPU award or project from the HSC and on the phytoextract in combination with the plant with the stem cells, so that, as you know, that there is a science which is Hikmat science, or there are plant-based science that are being used from the centuries, and they are currently being also used in locally also. So the plants have flavonoids, terpenoids, and other phenolic compounds. They are used for the inflammation, for the coronary diseases, and for other diseases. So we are using these phytochemicals or extracted from the different plants, mostly from the northern area of the Pakistan, and then we are using in combination with the stem cells, and then we are using it for the cure of the basically osteoarthritis. And we have seen that one of my PhD students is getting a degree on the Monday, and his work is also on the phytoextracts and the stem cells, and the work is very promising. And we have seen that the basic purpose to use this is that plants had anti-inflammatory as well as antioxidant compounds. The stem cells also has anti-inflammatory and antioxidant properties. But if we merge these two properties together, then we can increase it many folds, and then we can use it, transplant it. Because in any disease, there is a procedure of inflammation. The liver disease, there is inflammation. In the cartilage disease, as you know, that there is inflammation, swelling, and pain, and because there is a damage, there is a production of reactive oxygen species there. So when we transplant the cells into that disease or inflammatory conditions, then there is a chances that the stem cells get wasted, they get damaged, and they cannot survive there properly. So we provide the stem cells with these antioxidants, anti-inflammatory compounds, and then we transplant it into the disease place. So there is a more chances and survival of the stem cells there. So after that, we are also working on nanoparticles also. We make animal model, and then we transplant the cells, and then we do different molecular biological assays to check whether the disease is reverted back, there is a reversal of the disease, there is a regeneration of that specific tissue or organ or not. This is one of the images showing this is from the publication in which we use the adipose stem cells, which were differentiated into chondrocyte, which are the cells of the cartilage. And then we have seen that the differentiated chondrocyte transplanted and ADSs transplanted, these were more close to the normal as compared to the OA, which is osteoarthritis. And we did the histology also, and the other Wakitani and other Mannkin stores, and we saw that there was a regeneration in the transplanted animals. We have, I will also discuss in the later slide. As I told you that there is a placenta around the baby, and there are different layers of the placenta which are covered by membrane, which is called as the amniotic membrane. This amniotic membrane is very much important structure around the placenta. It has very high tensile strength, and it has very important growth factors also. So it can help, it can be used as the scaffold for the culturing of the stem cells also. So we have used that amniotic membrane also for the culturing of the stem cells and even the differentiation of the stem cells. And we have published it internationally, and around the 4.3 impact factor. And then we have seen that it helps in the culturing or growth of the stem cells very well. So this is the image from that publication that shows that the plastic surface also supported the growth of the cells, as well as the human amniotic membrane. It supported more than the plastic surface. as in previous slide, I tell you that amniotic membrane is used for the culturing procedure, but from many years, around 1920s, 1940s, this amniotic membrane is being used for the ophthalmology procedures. Doctors or ophthalmologists use that amniotic membrane on the eye of the patient, and it helps in the reduction of the inflammation in the eye of the patient, as well as for the cure of some kind of diseases there. So we are in collaboration with LRBT hospital mostly, but many other hospitals like Medical Hospital, and there was other hospital, New Hospital, Adil Hospital, Avicenna Hospital, etc. They take the amniotic membrane from us, and we take it from the patient. We do all the screening, viral screening of the patient, that there is no virus in the mother. Then we make the amniotic membrane, and then we freeze it at -80°C. And when there is a demand, the patient comes, and then he takes the amniotic membrane from us, and then the doctor transplant on the patient eye, and there is some help to the patient. We do it with the no cost to the patient, and we use our facility and our all resources for the benefit of the masses. Other thing which is being done with the help of this with the help of this amniotic membrane is the limbal stem cells transplantation. What is limbal stem cells? There is a layer of limbus tissue around this cornea of the eye, and it helps in the movement of the cells from this part on the cornea. But when there is a damage on this limbal structure, then the cells from this adjacent layer get over the cornea, and then that cornea is covered, and there is opacity on the lens of the patient. So what the doctor do, if the one eye of the patient is having this limbal stem cells deficiency and it has opaqueness on it, then what the doctor do, they take the limbus tissue, very small limbus tissue from the healthy eye of the patient, and then they provide us, and then do the it has the limbal stem cells in it, so we can isolate the stem cell, limbal stem cells from it, and then we grow it in a large number in certain stages. We characterize it, and then we provide it to the doctor in the form of an injection, and doctor put it back into the damaged eye of the patient, and that stem cells will help in the healing of that eye. And we have seen that there was a cure, and there was regeneration, and there was a very good vision improvement, acuity improvement in that damaged eye of the patient. So whether it was a burn patient, alkali burn patient, and acid burn patient, we have transplanted on a 10 years child way back many years ago also, that he has a damaged eye due to some car battery acid, and his eye was improved very well, and the acuity was improved also. So we also provided, but mostly the doctor take the amniotic membrane from us, and we have a bank of that amniotic membrane. This picture just shows you the structure and the place from where we take the amniotic membrane, and we then, after procedure, preserve it at -80°C in the frozen form, and then we provide it on the demand of the doctor. This is the image showing the pre-op and post-op of the patient having the chemical injury of the both eye, and you can see that the counting finger 1 and counting finger 1 was improved to the counting finger 5 meter. It means the patient was able to see or can count the finger from the 1 meter before the transplantation, and after that from the 5 meter distance he was able to count the fingers. This is also the same for the another procedure, and the VN was improved. This was the basic research, and the later on the clinical side in which we were providing the amniotic membrane to the different hospital. Now coming to the clinical side of the stem cells. So science is moving from the drugs to the personalized medicine. Means me is basically the source of my cure also. The cells inside me, which are the stem cells, they can be used any time of my life for the cure of many diseases in my body. There are countries, very advanced countries, even in India also, and in Pakistan, some international companies taking a lot of money. What they are doing, they are in India also and other countries, they are teaching people, they are giving awareness to the people that when there is a child delivery, then we are here, you donate your umbilical cord or umbilical cord blood to us. We will take a lot of money from you, and then we will freeze, or we will secure that, preserve that umbilical cord or your cord blood in for the lifetime. And when there is a need or any disease in your children, then he can use that stem cells, preserved stem cells, and take it from us, and then we can cure it. They are saying that this is the most precious gift that you can give to your children, because but it is a very costly procedure, and they use liquid nitrogen and other all the things on. They take it initially money from it, and on the monthly basis they take the money from the patient. But people are doing this very well, financially very good patients or person, they are doing this. But if we are not able to do that in our country, but we can help the patients also in another way. What is that procedure? We can make some good facility, a large facility, where we can isolate the stem cells, and then we can provide it in the bulk for the treatment of the patient. There are two sources of the stem cells. One is called as allogenic, and other one is called as autologous. Allogenic is from the other patient, other person. Autologous is from the myself only. So we can use allogenic source, like we can use umbilical cord. Umbilical cord is more like to fetus, so it does not have any immune rejection if we transplant into the patient. So we can use that umbilical cord, but if we are not willing, we want to use our cells only, then we can take the lipoaspirate, and then we can isolate the cells, and then we can provide to the doctors. And many clinicians, many surgeons, and many private clinics, they are saying that they are importing the stem cells from other countries, and many private companies, international countries are working evenly now in Pakistan, and they are charging 5 lakh, 10 lakh, 2 lakh, 3 lakh, many different basis on the patient pocket. So they are giving to the patient, and I'm not saying that all corrupt, they are all wrong, and they are all illegal in the sense that उसमें कुछ होता नहीं है. Maybe अगर किसी में होता भी हो, cells भी होते होंगे, transplant कर रहे हैं, and then उनमें improvement कुछ कहते आती है, but I'm not sure कि उसमें कुछ होता भी है कि नहीं होता है. तो अभी जो है हम ये प्लान हमारा, क्योंकि एक बेसिक रिसर्च का इदारा है, but we want to help the patient, and we want to move from the bench to the clinics. We want to help the clinic and to help the patient later on. So for that purpose, there are different authorities and different agencies. From that, we have to take the permission, etc., etc. For the research purpose, there is no need of that permission, but for the other purposes, on the clinical or the commercial purposes, we need some kind of permissions. So we are passing through that, and also there is a need of CGMP facility, which is called as the Current Good Manufacturing Practices Lab. It is a lab like the OT, operation theater of the hospital, that it doesn't have curves, and particle count is that much, and there is no contamination, bacteria, etc., etc., in the air, etc. so there is no chances of the any contamination to the cells, and we can provide the cells without any contamination to the different doctors. So we have in collaboration with the Burn Center also, and we have a very small setup there which is CGMP approved there, and we are culturing stem cells there, and we are providing for the research purposes only. I will mention that, and my colleague Dr. Azra, she is working on facial reconstruction and as well as the diabetic foot ulcers, and she is providing to doctors for the treatment of that disease. And one other project which we have done is also the treatment of the patient with the COVID-19 patient with the help of this umbilical cord stem cells. In the last slide, I will also show you another publication around the world. And also, thank you. इतना बोल रही हूँ. अच्छा, अभी इसको खत्म करते हैं. नहीं लग रहा था मैं तो 30 मिनट में खत्म कर दूँगी. अब ये बस ये शो कर रहा है that you have a CGMP facility where you can either culture the fresh cells and then provide it into the hospital, as well as you can grow the cells and then you can freeze the cells and then you can provide it to the hospital for the transplantation purposes. These are the different diseases on which around the world they are transplanting the cells, like Alzheimer's disease, Parkinson's disease, spinal cord, cartilage injury, heart disease, spear burns, and diabetes also. They are being currently in clinics for the treatment of the diseases. And one is for the baldness, like they take the stem cells or fat tissue and then in the clinics, and then they in the tube they digest it and spin it, and then adipocytes are also mixed. Many aesthetic clinics are doing this, and they transplant it for the facial reconstruction. If there is some kind of किसी के यहाँ पर गड़े पड़े हुए हैं, यहाँ पर जैसे हमारे बुढ़ापे के साथ साथ हमारे यहाँ पर भी पड़ जाते हैं, तो उसके अंदर आप फैट का करवा लें, तो जैसे और चीजें आप सबको पता है, तो वो करवा के फिर फ्लफी हो जाता है, आप यंग लगते हैं, और बाल नहीं है तो आप बालों पे लगवा लें और बाल भी थोड़े से आ जाते हैं. बर्न पेशेंट सीवियर पेन में होते हैं, इनके लिए हमें मतलब वो तो एक खूबसूरती और उसके लिए है, लेकिन अगर आप इसकी बीमारी और पेन को आप हेल्प कर सकें, वो उससे बढ़ के कोई काम नहीं है. तो आप बर्न पेशेंट्स के लिए आप उसको दें और उसके आप स्किन पैच या एम्नियोटिक मेम्ब्रेन के पैच उस पर लगा दें, क्योंकि जो ग्राफ्ट आती हैं बाहर से वो इतने महंगे होती हैं. इतना सा ग्राफ्ट जो होता है कैडावेरिक स्किन का वो लाखों रुपए में होता है. अगर किसी का बहुत बर्न हुआ है तो हम जैसे कंट्री उसमें वहां पर आप उसको नहीं दे सकते हैं, वो सर्वाइव नहीं करता. बाहर के मुल्क में 50-60% बर्न पेशेंट भी सर्वाइव कर जाते हैं फैसिलिटीज की वजह से, अमाउंट ऑफ अच्छी मनी की वजह से. यहाँ पर नहीं करते हैं, तो हम अगर उनको हेल्प कर सके तो हमारा एक हमें बहुत पेशेंट के लिए बहुत अच्छा होगा और हमारे लिए भी अच्छा होगा. डिफरेंट डिजीजेज है जो हेमाटोपोइटिक स्टेम सेल्स या बोन मैरो स्टेम सेल्स जो है उसमें ट्रांसप्लांट किए गए हैं, या उसका डाटा है अराउंड द वर्ल्ड. एंड देयर इज ए लेडी somehow old now, Lisa Roy, and she was diagnosed with some blood cancer, and she was treated with the help of the stem cells transplantation, and now mashallah she is recovered, and now she is the brand ambassador of the stem cells therapy in the India. These are some of the papers around the world. You can see that neural stem cells transplantation from the spinal cord injury. I will mention that I am in collaboration with Ghutki Trust Hospital, which is a very good non-hospital. Dr. Amir Aziz is the chairman, and we are planning to, inshallah, for the research purposes only, and we are planning to transplant the umbilical cord stem cell on the spinal cord injured patient as well as on the osteoarthritis patient. Adipose type stem cell, there is a knee injury or any sports injury or any, with the passage of time, all the women around in Pakistan, I think that they get the problem of these knees, and उनमें जोड़ों में दर्द होती है, घुटनों में दर्द है, वेट ज्यादा है, कुछ आपकी लाइफस्टाइल है, कुछ आपकी जेनेटिक में ट्रांसफर भी होता है, वो सब में प्रॉब्लम है. आप उसमें ट्रांसप्लांटेशन करवाएं, और बहुत से क्लिनिक्स ये काम कर रहे हैं, पैसे ले रहे हैं 2-2, 4-4, 5-5 लाख, और वो कह रहे हैं कि हम ट्रांसप्लांट कर रहे हैं. और वैसे एक को मैं जानती हूँ कि ना हमारे वहाँ पे कोलीग है, उन्होंने अपनी मिसेस का कहीं जिक्र आया था, वो कह रहे हैं बड़ी खुश हैं और उनको दर्द नहीं होती है, तो वो तो कह रही मेरी जिंदगी बदल गई है. और दूसरा एक HIV, आपको पता है कि HIV एक डरावना ख्वाब है जिसके बारे में हम बचपन से सुनते आ रहे हैं, लेकिन अब इसका कुछ कुछ ट्रीटमेंट्स आता जा रहा है। और इसमें से एक जो मेन ट्रीटमेंट जो किया गया है एक पेशेंट में, वो HIV के पेशेंट में पॉजिटिव पेशेंट था और उसका देखा गया कि वो नेगेटिव हो गया। और उसमें क्योंकि उसमें वो उसको कोई और ब्लड की बीमारी थी कैंसर की, उसके ट्रीटमेंट के लिए उन्होंने एक डोनर का जो है उसमें ट्रांसप्लांटेशन की, तो जिसमें जो CCR5 और 32 जो है उसकी हिमेटोपोइएटिक स्टेम सेल्स की उसमें म्यूटेशन थी, यानी कि वो उसके लिए था। तो उस पेशेंट में जब ट्रांसप्लांटेशन की गई तो देखा गया कि उस पेशेंट में रिमिशन होगी, क्योंकि जो रिसेप्टर है वही आपका जो एड्स का वायरस है वो जाकर अटैक करता है और सेल्स के अंदर ग्रो करता है। और ये पेशेंट एक है जिसमें अभी तक उसकी रिमिशन जो है वो देखी गई है। स्टेम सेल्स, मैंने कोविड-19 के पेशेंट की आपको बात की थी, हमने भी कुछ पेशेंट्स पे किया है, उनका डाटा भी लिया है। मतलब एक कह लें कि मॉडरेट रिजल्ट्स थे। कुछ आपको पता है कि सीवियर केसेस होते हैं तो उसमें तो कोई कार्डियक अरेस्ट वगैरह हो तो वो चांसेस सर्वाइव के नहीं होते हैं, लेकिन हमने ये देखा कि उनमें जो डिफरेंट इंटरल्यूकिन्स जो होते हैं वो काफ़ी उनमें चेंजेस आई और इम्प्रूवमेंट हुई है, लेस हुए थे एज कम्पेयर टू द कंट्रोल पेशेंट। ये एक रिसेंटली थी, BBC न्यूज़ में पढ़ती हूँ डेली, तो उसमें ये खबर पिछले दिनों मैंने पढ़ी कि पेशेंट है जिमी जिसको सिकल सेल एनीमिया की बीमारी थी, पब्लिश डाटा नहीं है बट एक फ्रॉम द न्यूज़ है। उसमें भी इसकी बीमारी थी और बहुत पेनफुल थी क्योंकि ब्लड में ऑक्सीजन को सेल्स मूव करवाते हैं, तो बहुत ज़्यादा प्रोसीजर से गुजरा, लेकिन फिर इसको कहीं पता चला कि वो कोई प्लानिंग कर रही है स्टेम सेल्स की और उसमें थोड़ी सी चेंजेस लाएंगे और फिर उसमें ट्रांसप्लांट इसने वो करवाया एंड नाउ ही इज़ वेरी हैप्पी। उसका पूरा एक आर्टिकल है, कोई पढ़ना चाहे मैंने नीचे लिंक पे दिया हुआ है। उसमें प्रोसीजर वो क्या था कि उन्होंने उसका बोन मैरो, इस बोन मैरो में जो सेल्स थे डिजीज थे दैट कैन मेक रेड ब्लड सेल्स सिकल सेल्स बना लेते थे, उन्होंने उसके बोन मैरो से जो है स्टेम सेल्स को आइसोलेट किया और वो जो है एक BCL11A जीन को जो है वो टर्न ऑफ कर दिया और जिसकी वजह से जो है वो उसमें कह लेंगे, अच्छा ये है stem cells were extracted, stem cells genetically were modified and stem cells were given back to the Jimmy and GD stem cells make the healthy fetal tissue। बेसिक आइडिया उसके पीछे भी ये था कि ये जो था ना BCL11 जीन जो था, ये जो क्योंकि कहते हैं ना फीटस में कुछ और तरह के जीन्स होते हैं, टर्न ऑन टर्न ऑफ होते हैं, तो ये जीन एडल्ट्स में आके टर्न ऑन होता है और जबकि जो फीटल जीन है वो और होता है, उन्होंने उसको वो वाला जीन को एक्टिव कर दिया कि वो वाला एक्टिव हो जाए और सेल्स बनाना शुरू कर दे नॉर्मल और मैच्योर सेल्स बनना शुरू खत्म कर दे। इस पे उन्होंने ये प्रोसीजर कर और उसको उसमें ट्रांसप्लांट किया एंड ये एक केस है जो कि उसको बहुत अच्छा है, वो खुश है अल्हम्दुलिल्लाह। थैंक यू वेरी मच। क्वेश्चन करना चाहते हैं? वैसे तो मेरे टाइम से ऊपर चली गई हूँ।
जैसे स्टेम सेल ट्रांसप्लांट कर लेते हैं तो उसका इफेक्ट कितने अरसे के बाद जो है ना वो नजर आना शुरू हो जाता है आफ्टर ट्रांसप्लांटेशन?
अभी कहाँ जाता है, ज्यादातर पेट में ट्रांसप्लांट किया जाता है वो ज्यादा 6 to 2 years तक फॉलो किया जाता है, असर नजर आना शुरू हो जाता है। लेकिन समटाइम ये होता है ना कि आप कुछ दवा भी खाते हैं तो फौरन तो आपको असर आता है, स्टेरॉइड होता है फौरन ठीक हो जाते हैं, लेकिन कुछ अर्से बाद उसका क्या इफेक्ट रहता है, आप ठीक रहते हैं कि नहीं ठीक रहते हैं, उसको देखने के लिए तो आपको ज्यादा अर्से तक उसको फॉलो करना पड़ता है। तो स्टेम सेल्स की जितनी स्टडीज हैं या कुछ हैं तो उसमें 2 साल तक वो कहते हैं कि आप फॉलो करें और वो पेशेंट को ऐसी स्टडीज भी हैं जिन्हें फॉलो किया गया और देखा गया है। तो डिफरेंट है, देखिए कोई भी चीज हर पे आके नहीं होती है, कोई भी प्रोसीजर दवाई भी नहीं है, हर एक नई चीज वक्त के साथ बदलती रहती है, लेकिन कुछ जो है एडवांसेस होती है, रिसर्च हो रही है, इंप्रूवमेंट्स आ रही हैं, जैसे इस पेशेंट में ट्रांसप्लांटेशन है और पेशेंट्स हैं तो किसी की हर एक की बॉडी का रिस्पांस डिफरेंट होता है, तो इसलिए वो उनके किसी में बहुत अच्छी इंप्रूवमेंट आती है और किसी में मॉडरेट आती है, किसी में नहीं होती है, तो मुख्तलिफ इसमें रीजंस होती हैं। आ रही है आवाज मुझे?
उनके शॉट्स होते हैं डिफरेंट, लाइक 3 months और 6 month के बाद एक शॉट, फिर अगला शॉट, फिर अगला शॉट, बट अगेन द थिंग इज कि इंटरनल बॉडी अगर एजिंग की तरफ जा रहा है और वो अपनी टीलोमेरेज एक्टिविटी को लूज करता जा रहा है, तो अल्टीमेटली जो आपने सेल ट्रांसप्लांट भी करवाया उसकी भी एक्टिविटी लूज होती जाती है। तो वो रेगुलर शॉट्स फिर रिक्वायर्ड है, पर ऑब्वियसली रिकमेंडेड रेगुलर शॉट्स भी 4-5 ही हैं, इससे ज्यादा तो कोई भी फिलहाल नहीं कर रहा। तो उसकी तो यस,
मतलब है कि
वो रिवर्ट बैक होगा, लेकिन कुछ अरसे के लिए इसको आप रोक सकेंगे ट्रांसप्लांटेशन के
जैसे
थ्रू।
आप ड्रग्स भी लेते रहते हैं, मल्टीवाइटामिन भी ले रही हैं, तो इस केस में हम बार बार तो ट्रांसप्लांटेशन देखें वैसे नहीं करवाते हैं। जैसे सना ने कहा कि जैसे आप बुटॉक्स भी लोग करवाते हैं, फेस के बार बार आते हैं आपके रिंकल्स को खत्म कराने के लिए, तो वो स्टेम्स भी आपने रिवाइज किए, कोई वो मतलब कोई ऐसी चीज तो नहीं है कि कभी खत्म ही नहीं होंगी, आपकी इंप्रूवमेंट होती है, क्योंकि वो पैराकाइन इफेक्ट कर रही है, तो उसने अपनी आपकी बॉडी में से रिक्रूट करना है ग्रोथ फैक्टर्स को, तो उन्होंने ज्यादा काम, दो इसके बारे में थ्योरीज है, या तो वो डिफरेंशिएट होती है और अपना काम करती हैं, या ये पैराकाइन इफेक्ट है, तो जिसकी वजह से पैराकाइन की वजह से रिक्रूटमेंट हुई और वहां पर एक बॉडी का रिजनरेशन स्टार्ट हुआ। लेकिन अगर एक ऑर्गन ट्रांसप्लांटेशन की तरफ जा रहा था और आपको बहुत पेनफुल थी और आपने करके हो गए, तो वो एटलीस्ट आप 2-4 साल बाद भी आपको एक और इंजेक्शन लगाना पड़ जाता है, तो इस बर्थ के आप उसमें तमाम में अपना गुजारा आपका जो है वो अच्छा हो गया।
जो आपने रिवर्ट बैक वाली टेक्निक बताई है कि आप सोमैटिक स्टेम सेल को लेकर के और देन उनको सोमैटिक सेल्स को लेकर के देन उसको रिवर्ट बैक कर रहे हैं स्टेम प्लूरिपोटेंट स्टेम सेल के अंदर, तो उसको क्या जरूरत थी जब हम स्टेम सेल ले सकते हैं?
देखिए इनकी लिमिटेशंस हैं, एडल्ट स्टेम सेल्स पे हम काम कर रहे हैं, उसका तमाम
तो ये ज्यादा लेबोरियस नहीं हो जाएगा कि पहले रिवर्ट बैक करें देन उसको डिफरेंशिएट करें?
बहुत बढ़ जाती है, आपका एक स्टॉक बन गया है, आप लाइफ टाइम उसको स्टॉक रख सकते हैं, आपने किसी भी लीनेज में उसको डिफरेंशिएट करना है, बाद में आपने उसको
तो सेम प्रॉपर्टीज होती हैं स्टेम सेल वाली उनके अंदर भी जो, लेकिन या डिपेंडिंग ऑन द फैक्टर्स वी आर गिविंग दोस।
मैं बहुत इस पे काम हो रहा है और एक अच्छा है।
There is an added property, जब हम stem cells को IPCs में convert करते हैं तो they can be used specifically for the personalized medicines। और नॉर्मली ये होता है कि हमारे पास जब हम एलोजेनिक की तरफ जाएं, हमारे मुल्क में और जगह पे भी एलोजेनिक इज कम्प्लीटली बैन। होता हमें सिर्फ और सिर्फ ऑटोजेनिक ऑटोलॉगस जो ट्रांसप्लांटेशन है वो अलाउ करता है, दूसरी कोई अलाउ नहीं करता, वो भी चंद लैब्स को ही अलाउड है, भी बाकियों को तो परमिशन भी नहीं है। तो जब हम एलोजेनिक से हटना चाहेंगे और मल्टीपोटेंसी को भी खत्म करना चाहेंगे ताकि एक कंप्लीट रिकंस्ट्रक्शन की तरफ हम जा सकें, तो उसके लिए आईपीसीज ही हैं जो एक इंडिविजुअल से लेके आप उसके अंदर, क्योंकि जो एचएलएज हैं और जितने भी सरफेस मार्कर्स हैं रिलेटेड टू पर्सन वो वहीं के वहीं इंटैक्ट रहते हैं और स्टेम सेल्स की प्रॉपर्टीज भी आ जाती हैं। वो आप समझ लें कि
पर्सनलाइज्ड मेडिसिन, जैसे मैंने कहा कि आप साइंस मूव कर रही है,
Thank you very much for a beautiful talk and very informative. I have a question that when you apply the phytochemicals, so it is a great chance of contamination into the stem cells. So what criteria you adopt to for the introduction of these phytochemicals?
Yes, but then how that we isolate the phytochemicals, then we dissolve in special kinds of medium like methanol extracts है, like water में dissolve है, किसी को है, उसमें हम जब उसका solution बना लेते हैं extracts का, हमने तो उसको हम किसी भी बैक्टीरिया से remove रखने के लिए हम उसकी filtration करते हैं 0.22 syringe filter से, और इस तरह से, और फिर जब हम stem cell culturing करते हैं और वो हमारा यूवी वगैरह सब ऑन करके पहले उसमें, और फिर हमारी स्टेरिलाइजेशन प्रोसीजर होती है, तो हमारे कभी फाइटोकेमिकल एक्सट्रैक्ट की उसमें कॉम्बिनेशन से कभी कंटामिनेशन नहीं आई है। हमारे जो ज्यादा चांसेस हैं आते हैं क्योंकि हम हॉस्पिटल से सैंपल लेते हैं, हम एम्ब्लाइकल कॉर्ड भी लेते हैं, थोड़ा सा उसका एक्सपोजर होता है, या लैप्टोस्पिरेट लेते हैं तब भी वो सही तरह से नहीं, या जिन दोनों में ह्यूमिडिटी के चांसेस बहुत होते हैं, जुलाई अगस्त दिनों में फंगल ग्रोथ वगैरह उन दिनों में ज्यादा आती है, तब हमारे इंक्यूबेटर्स में थोड़ी हमें कंटामिनेशन वो नजर आ जाती है कि वो आपके सेल्स उतना शुरू हो गया, बैक्टीरिया ग्रो कर रहे हैं, फंगस के आपके स्पोर्स नजर आ रहे हैं। लेकिन जो नॉट ओनली द फाइटोकेमिकल एक्सट्रैक्ट, वी यूज मल्टीपल ऑफ अदर कंपाउंड्स, pharmaceutical compounds, vitamin E, alpha lipoic acid, and the curcumin and other different compounds, pure compounds also, and then we mix it in an environment in a suitable solution, and then we syringe filter it, sterile it, it's okay, there is no chance of contamination with that.
बहुत शुक्रिया।
Thank you so much, ma'am. And thank you so much for joining us at very short notice. Thank you, ma'am. Thank you for joining us at very short notice, and it was very.
It's really an honor for me, and I'm happy to visit the virtual university.
Thank you so much.
Listen the name of your university, which I was.
Okay.
Thanks to Azam that he gave me chance.
So nice of you. Ladies and gentlemen, let's take a tea break, and we'll resume the second session at 11:45. Welcome back. Ladies and gentlemen, our next presenter is Dr. Sumera Anju. She is Assistant Professor at Department of Biotechnology, Canaid College for Women, Lahore. She is a young and enthusiastic early career researcher in the field of nanobiotechnology and plant biotechnology. She always remained a brilliant student during her education career and also proved herself as an excellent researcher in a very short period of time. She has published many research articles in internationally recognized journals with an accumulative impact factor of 112 and more than 550 citations. She has synthesized a large variety of metallic and metal oxide nanoparticles and evaluate their biomedical. Today, he will deliver her talk on an effective simulator of plant-mediated biosynthesis of nanoparticles and their biological applications. Welcome, Dr. Sumera. Please proceed.
Assalamu alaikum. First of all, I would like to say thanks to Virtual University for inviting me on this talk. And I am feeling honored because my all senior faculty from Canaid College is sitting here. Thank you so much. तो start करने से पहले audience, I have distributed a questionnaire. This is just to check your basic background about the nanotechnology. Please don't Google it. Just circle the correct option. And after the lecture, we will again solve this questionnaire to check the either you have gained anything from my lecture. That is usually the basic questions of nanobiotechnology. You have 5 minutes for the questionnaire, then we will start the talk. Thank you. Gee, I think all the audience have done with it. जी 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 कर रहे हैं। जी, जी, Today I am going to talk about a research-based topic, that is light and effective stimulator for plant-mediated synthesis of nanoparticles and their biological activities. This is basically a research-based topic, but obviously before going into the details of this topic, I will briefly give a short introduction of nanotechnology, what is nanobiotechnology, what is the classifications of nanomaterials and why nanomaterials are important, what are the sources of the nanomaterials, what are the properties of the nanomaterials, what are the journal applications of the nanomaterials, and then what are the biomedical applications of the nanomaterials. And at the end, we will going to discuss the specific topic of research, light as an effective stimulator for plant-mediated synthesis of nanoparticles and their biological activities. I would like to start the talk with the famous quotation of Sir Eric Russell. It's always the small pieces that make the big pictures. This quotation so beautifully clarify the concept of nanotechnology. It's really the small things which are going to the next big things. So nanotechnology is basically derived from the word dwarfism, like nano means small. So basically the all beauty of the nanotechnology lies in its small size. That is the main feature of the nanotechnology. And in the current era, the nanotechnology has revolutionized the every field of science, engineering, and technology. And it has impacted the field of science to such an extent that it has changed the thinking of the researchers. The researchers have started to thinking in a new ways that how these medicines can be targeted to a specific target. There are so many biomedical applications of nanotechnology in life sciences. So next we start करते हैं इसका sorry. What is nanotechnology? First, this is a pure specific definitions of nanotechnology. I'm not going into the reading of this definition, just giving you an idea. Basically, जो nanotechnology field है, in this field is an interdisciplinary field in which we coined the two basic sciences. One is the science and other is the engineering, to fabricate different kind of devices and also to synthesize different kinds of materials. And the materials which are synthesized by using the nanotechnology are then characterized and then they applied for their applications. So basically nanotechnology is the synthesis and fabrications of different kinds of devices and materials which are then characterized and used for various applications in different fields of sciences. But the materials either we are synthesizing, how we can say that it fall in the category of the nanotechnology, or either it relates, it's a nanomaterial. Then basically the main key point that कि कोई भी जो मटेरियल है वो नैनो की फील्ड में फॉल करता है, तो it is that it should be at least in one dimension in nanometer scale. And what is about one dimension? One dimension means to say, if you have synthesized any nanomaterial, either it should be in its length, in nanometer scale, either it should be in width of nanometer scale, or either it should be its height in nanometer scale. In any one parameter, it should be in the nanometer scale. It's mean if you have a rod that is 1 meter long, but it has a diameter in nanometer scale, then it falls in the category of the nanotechnology. It's basically a nanomaterial. So then the main peculiar point is that it should fall in nanoscale in at least one dimension. Now, what is nanoscale? You all know that 1 nanometer is equal to 10 raised power 9 meter. That is a very, very small. So you can have an idea about the nano things from this figure, like these particles of the sands are not the nanomaterials. They fall in the scale of millimeter. Pollens from the plants in micrometer. Red blood cells are even very small, but not they are in the scale of the nanomaterial. Similarly, the bacteria, microorganisms are not in the nanometer scale. Yes, the viruses started from the nanomaterials. These are in the dimensions of the nanoscale. Similarly, some proteins are there which are in nanometer scale. DNA is approximately 2 nanometer in diameter. Similarly, the carbon atoms, fullerenes are also in the nanometer scale, and the atoms are also in the nanometer scale, like carbon, hydrogen, oxygen, then their protons, electrons all fall in the nanometer scale. Now, as I already mentioned, nanotechnology is a interdisciplinary field. Like कुछ लोगों का ये कॉन्सेप्ट होता है कि जो जिस फील्ड में काम कर रहे होते हैं, लाइक वी आर फ्रॉम द लाइफ साइंसेज, वी थिंक दैट कि नैनो टेक्नोलॉजी इज प्रॉपर्टी ऑफ द लाइफ साइंसेज. All think is all about nanobiotechnology. And if you belong to the physics, they said nanotechnology belongs to the physics. It's the domain of the physics. And the chemist said nanotechnology is the domain of the chemist. But it's not true. Basically, nanotechnology is an interdisciplinary field. और इसके अगर हम basic concept या history की बात करें, तो basically the origin of the nanotechnology was just started in 1969. In 1969, Sir Richard Feynman, which is called as the father of the nanotechnology, he gives a idea of small things. Sir Richard Feynman basically delivered a lecture in University of California with the title, "There is a plenty of room in the bottom," in which he gave the idea of small things, but he didn't use the word of nanotechnology. At that time, the idea given by the Sir Richard Feynman didn't gain such popularity or didn't get much attention until in 1986. Sir Eric Drexler published a book with the name of Creation of Engines, in which he discussed the nature. And he correlated the small things with the nature, like Allah has created us, our cells in nanometer scale, and our organelles, mitochondria, Golgi bodies, they are in the nanometer scales. And he said Allah has beautifully created these organelles, and they are performing so many complex functions. Then humans can also engineer such types of materials which can perform different kinds of big functions. So basically, he was the first, Sir Eric Drexler. He used the word nanotechnology. He has introduced the word nanotechnology, but he was a philosopher. He was not the scientist. So he just gave the idea of nanotechnology, and after 1986 till 2022, almost there are 36 years, only 36 years, and nanotechnology has boomed every field of science. It's a big market, and it's the largest market. Currently, I was seeing a paper in which that was published on the market value of the nanotechnology. Like in European countries, it's going to be $3 trillion industry in 2024. And in Europe, it's going to be $2 billion industry in 2024. And in Asian countries, it's going to be $5 billion industry in 2024. It's mean कि जितनी भी अभी जो current industry चल रही है, उसमें से सबसे ज़्यादा जो high performing industry है, that is nanotechnology, and it has just boomed in 35 years periods of time. That is a very short period of time. So basically, it's belong to all fields of sciences: chemistry, physics, bio, medicines, computer sciences, electronics, and it's basically an interdisciplinary field, and it's an individual subject. It's not a part or property of any other subject. Now, we said that in nanotechnology, we devices different kinds of devices, manufacture different kinds of devices, or either we synthesize different kinds of materials. Now, what kind of materials we can synthesize by using the nanotechnology? So basically, nanomaterials have been classified into three main categories. One is called as the organic organic type. Second is classified as the inorganic type, and third one is called as a hybrid. If we further divided them, then the organic nature belong to polymeric nanoparticles, liposome or lipid-based nanoparticles, dendrimers. These are belong to the organic nature. These kinds of nanomaterials basically are used in nanomedicines for drug loading. Like dendrimers, you can see have as a branch structure. So due to its branching, it can load a variety of drugs, and a high quantity of drug can be loaded. Next, the inorganic nanoparticles are usually metal-based nanoparticles. Metal-based can also be classified further into two classes, like metallic nanoparticles are like silver nanoparticles, gold nanoparticles, titanium nanoparticles, copper nanoparticles, and also the metal oxide nanoparticles. Metal oxide like zinc oxide nanoparticles, titanium oxide, copper oxide. These are all of the inorganic nature and metal-based. The other class with the inorganic material is carbon-based nanomaterials. The carbon-based nanomaterials are further classified into carbon nanotubes, which can be either single-walled carbon nanotubes or the multi-walled carbon nanotubes, which are further used in various applications of electronics and also for biomedical applications. And the quantum dots. The quantum dots are really, really small and the most amazing type of nanomaterials, which are although currently manufacturing is very expensive, but they have been commercialized due to their quantum confinement principle. You all might be seen a car that automatically change the colors of the surfaces, in which the quantum dot technology has been used. Just to give a push of light or a source of light, the color of the car is automatically changed, either red, green, white, in which the materials used are the quantum dot materials. So due to their excellent optical properties, they are currently using in different types of devices, surfaces coating, and diagnostic imaging. So this is brief, what are the classification of nanomaterials? Now, why nanomaterials? This is the main thing, why nanomaterials are so much important, why we study nanotechnology. So basically, the nanomaterials, जैसा कि अभी हमने discuss किया है, they are important. The first property है, that is their size. They have very small size, and anything we all know that if it has a small size, then automatically its surface to area volume ratio increases. As you cut down and then become to a smaller level, its surface area become increasing. So main attractive जो property है nanomaterials की use करने की, that is first of all their small size, which correlates to the high surface area to the volume ratio. And when it increases the high surface area to volume ratio, then you can load a lot of drugs and other different kinds of materials can be loaded, can be surface functionalized, and they can be more reactive as compared to their bulk material. This is the major one. Other than that, why nanomaterials are important? They have unique thermal properties, like thermal properties, thermal conductivity. They have very low momentum and low size of the nanomaterials. Therefore, they diffuse heat so rapidly, and therefore they are having extra unique thermal properties and being used in different diagnostic purposes. They have the unique optical properties rather than their bulk material, like they have unique reflection, diffraction, absorption, transmission properties, which are basically using in diagnostic imaging, bioimaging में use हो रही हैं. इसकी ये property जो optical property है. Next is the mechanical property. They also shows the unique mechanical properties. The mechanical properties are like their hardness. They differ in hardness. They differ in elasticity. They are so tensile. They are lighter in weight, and they can be so thin in structure that you can craft thinner weight or lighter weight aeroplanes in space energy. They are using this property. Similarly, they are using this mechanical property to coating different substances with the lighter weight, like in automobile industry, they are coating with the nanomaterials to lighter their weights and to reduce the fuel consumption and to increase the speeds of the automobiles. Next is the unique same electrical properties. Some of the nanomaterials are conductors, some are semiconductor, and some are insulators. You can modify or you can manipulate the electrical properties of the nanomaterials by different techniques, and you can use in different applications. And the last one is the unique property is the magnetic properties. Some nanomaterials are ferromagnetic, means they are completely magnetic, and some are diamagnetic, either they are repelled by the magnetic field, and some are paramagnetic, they are attracted by the magnetic field. So by manipulating these magnetical properties, you can use them in different electronic devices like nanochips, nanobarcodes, nanobiosensors. So these all are the unique properties of the nanomaterials, that why we prefer nanomaterial than the bulk materials. Next, what are the sources of nanomaterials? The sources of the nanomaterials, basically three types of sources. And one is the natural resources that is created by nature. And the second one is the incidental, or you can say that accidental sources. And the third one are called as the engineered or intentionally generated nanomaterials. In natural resources, if we talk, then our blood is the best example of nanomaterial. Milk is the best example of nanomaterials. Is the suspensions of different kinds of nanomaterials. Similarly, silk produced by the silk mite, chalk by formamine, all are the examples of the naturally occurring nanomaterials. And if we now talk about the accidental nanomaterials that are not created by our intention, like mining, like different industrial processes, different waste from the industrial processes containing the heavy metals, which are then accumulating in our environment, contaminating our underground waters, and you can air, atmosphere contamination, or different industrial processes like grinding, corrosions of the metals are generated the fumes or dust of the nanomaterials, which are not created by intentionally. And the last one is the intentional. This is the main class in which we researchers are working. These are also called as the engineered nanomaterials because they are made by intentionally by the human beings, and different kinds of nanomaterials are currently have been synthesized and being synthesized by using different methods of synthesis that we are going to discuss in the next slide. Now coming towards the what are the roots of synthesis of nanomaterials, that how we can synthesize the nanomaterials. So basically the synthesis of the nanomaterials can be carried out by using two approaches. There are two main approaches. One is called as the top down and other is called as the bottom up approach. As you can see in the figure, in this one, in the top-down approach, you use the bulk as a starting material, and then you cut down, and comes into the nanoparticle level. While in the bottom-up approach, you use the starting material lesser than the nanometer scale, like atoms. Atoms are smaller than nanometer scale, and then you get assembly of them into the clusters and then created into the nanoparticles. So basically, these two main approaches are used for the synthesis of the nanomaterials. अब ये जो हमारे पास two main approaches हैं, either bottom up or top down. Then by using these two approaches, different methods have been formulated. Like there are three main methods that are used for the synthesis of the nanomaterial. One is called as chemical synthesis or chemical methods. Other one is the physical methods, and the third one are called as the biological methods. If we specifically talk about the chemical synthesis, then in chemical synthesis, we used the metal precursor salts in which you want to synthesize the, let's suppose, nanoparticle, which you want to synthesize the silver nanoparticle, let's suppose. Then you have to need a metal precursor salt that contain the silver, like you can use silver nitrate, you can use silver chloride, which containing the silver ions. And then you have to take another chemical which should reduce the silver ions from that solution, and that reagent that is using for reduction is called as a reducing agent. And different kinds of reducing agents are being used for reduction of different kinds of metals or different kinds of materials are currently being used. And after reduction, the nanoparticles have been synthesized. Then you need an extra stabilizing or capping agent to cap or stabilize the nanoparticle, because the nanomaterials or nanoparticles produced by the chemical methods are not capped until you have not capped or stabilized them by adding an other extra chemical. So basically, chemical methods are not the preferred one methods, because they involves a lot of chemicals that further contaminate our environment, and also we cannot use them for biomedical applications, because we are introducing these nanomaterial into our living systems, either animal models or either human beings. Then they are not recommended one nanoparticles that are synthesized from the chemical method. But अब ऐसा भी नहीं है कि फिर chemical method का कोई use ही नहीं है. There are uses of chemical methods like chemical synthesis, yield very refined nanoparticles that can be used in electronics, that can be used in construction industry, that can be used in food packaging, but we cannot introduce them into the living system or biological applications. The different types of chemical methods are currently in use, like co-precipitation method, one of the simplest and the basic method, one-pot reaction for synthesis of the nanoparticles. The other famous method is the hydrothermal method, and the most common method is the sol gel method, that is the most efficient in terms of its yield and in terms of the assembly of the nanomaterials. The next class is the physical methods. In physical methods, as the name shows, that we didn't use any kind of chemical, but we use the physical equipment, like we use the laser light, we use a source of energy to evaporate the metal. Like different kinds of physical methods are currently in use for the production of different kinds of nanomaterials, like laser ablation method is one of the most famous method that is currently used for physical generation of the nanomaterials, in which we use a pulse of laser light to evaporate our target metal. And once it's evaporated in the form of atoms, and these evaporated vapors are then collected into a cold finger and then scrap out from this cold finger, or you can say from the substrate. The main advantages of the physical method is that you can produce nanomaterials in bulk amount, but assembly is not controlled. They are not well assembled, they are not uniform in their size, but yes, you can synthesize in the bulk. Therefore, if you want a controlled synthesis of the nanomaterials, you have to control different factors. We are going to discuss in the next slide. The third methods are called as the biological methods. Biological methods are also called as green synthesis of nanomaterials. So green synthesis of nanomaterials is not basically the synthesis only from plants. Okay. This is not the synthesis only from plants, as we generally assume that greener plants and only the green synthesis. So therefore, green synthesis is not the only synthesis from plants. This is the synthesis from all biological sources. And there are mainly two types of biological sources are used for green synthesis of the nanomaterials. One is the main class microorganisms like bacteria, viruses, fungi, which contain a lot of oxidoreductase enzymes and different proteinases, which can cause the reduction of the metal ion from the precursor salts. And the other main class is the plant. You can use phytochemical enriched plant extracts can be used for the synthesis of the nanomaterial. The phytochemical rich plant extracts can be any medicinal plants which contain rich amount of phenolics or flavonoids or alkaloids or other kinds of secondary metabolites, which basically can act as reducing agents. Now, currently, the biological methods of synthesis and specifically the plant-mediated synthesis of nanoparticles is currently being more practicing technique for synthesis of the nanoparticle, especially for biological applications, because they didn't contain any hazardous chemical. on their surface. And the one major property or the one major point जो कि plant mediated synthesis का है, that is you didn't need any external capping or reducing agent. You just have to take the plant extract, and there is also no boundaries for the plant extract. You can take the leaf extract, you can take the stem extract, you can take the root extract, you can take the seed extract. Just you have to verify either it contain enough amount of the phytochemicals that can act as a reducing and stabilizing agent. If yes, this plant contain the enough phytochemicals that can cause the reduction and capping of the nanomaterials, then yes, of course, go for it. And this is the simplest method of synthesis, and these are nanoparticles produced by the biological methods are knives you can use as such into the biomedical applications. Now, I have discussed in the previous slide what is the green synthesis of nanomaterial that basically involves the two main routes, either from microorganisms and either from plants. And basically, जो green synthesis है, green synthesis को हम बहुत accurately term में देखा जाए तो anything not in the field of the life sciences, anything generated in chemistry, in physics, in electronics, if it follow the 12 set of principles of green chemistry, then it falls in the category of the green chemistry. These 12 rules of the principles were given by American Chemical Society, in which there are set of 12 rules कि either जो भी methodology adopt की गई है उनकी synthesis में वो eco-friendly है, वो safe है, बायोप्रोडक्ट सेफ हैं, जो मेथोडोलॉजी यूज की गई है वो कॉस्ट इफेक्टिव है, लेस सोर्स ऑफ एनर्जी यूज कर रही है. इफ दे फॉलो ऑल 12 सेट ऑफ प्रिंसिपल्स आइदर इन द क्रिएशन ऑफ इलेक्ट्रॉनिक डिवाइसेस, केमिकल डिवाइसेस, और आइदर इन द क्रिएशन ऑफ द नैनो मटेरियल्स, दे फॉल इन द ग्रीन केमिस्ट्री. बट इन case of nanotechnology, we specifically talk about green chemistry from two sources: organisms, microorganisms, and other one is the plant. Now, these both, as I already told you, the plants contains various phytochemicals. So basically, जो phytochemicals हैं, like alkaloids, phenolics, flavonoids, every plant contains certain amount of phytochemicals and different variety of phytochemicals. These phytochemicals are actually acting as a reducing agent, plus also acting as a stabilizing agent. There, it means you have no need to add any chemical agent for reduction of the metal ions, and you also need not any capping or surface agent to cap or stabilize the nanoparticles. By using the green methods or green synthesis of nanomaterials, you can synthesize the controlled or engineered nanoparticles of your own choice, of your own wish. You can engineer, you can formulate your types of nanomaterials by working or by controlling some factors. Which factors affect the synthesis of the nanomaterials? Like temperature. Temperature affects the synthesis of the nanomaterials. It affects the morphological characteristics of the nanomaterials. If you are using the same plant source, same molarity of the stock solution of the metal precursor, and one synthesis is on 30 degrees centigrade temperature, and other is the synthesis on the 60 degrees centigrade temperature, and other is on, let's suppose, 90 degrees centigrade temperature, temperature will surely affect the morphological properties. They will differ in their size, they will differ in their shape. So it means the temperature is one of the most factor you can optimize the temperature for your own choice of plant. जो भी आप plant पे काम कर रहे हैं, आप उस पे उसका temperature optimize कर सकते हैं to attain the desired or acquirable sizes. The second factor which affect the synthesis of the nanomaterial is the pH. You can change the pH, you can optimize the pH. pH, most of the time, the lower pH have been seen that they impacted the smaller size created the smaller size nanomaterials. If you increase the pH, then the sizes of the nanoparticles also increases. Similarly, you can modify the incubation period. Incubation period mean to say if you are going to synthesize, let's suppose, silver nanoparticles, and you have prepared the 1 millimolar stock solution of, let's suppose, silver nitrate, and you have prepared your plant extracts of the leaf of any plant, which is, let's say, 10 gram per 100 ml, and you are mixing them in a definite ratio, like 1:1, 1:2, 1:4. This is all your own optimization. You have to go through a series of optimization for synthesis of the required size, type, or morphology of the nanomaterials. So then you can also change the incubation time. If you give 10 minutes, the sizes of the nanoparticles may be differ to those 30 minutes incubation time. then 150 minutes incubation time. So incubation time basically increase the contact time, which increases the rate of reduction. And sometimes the incubation time also negatively impact the synthesis of the nanomaterials. They reverse back the oxidation reduction reactions, and the nanoparticles generating are not stable. Then again, you have to optimize the contact time according to your plant, according to your type of the nanomaterial. And the last one is the centrifugation. Centrifugation timing also matters. Either you are speed and timing giving for a larger time or shorter time, larger nanoparticle will be sedimented or the smaller nanoparticles will be sedimented. Now, my area of specialization is plant-mediated synthesis of nanoparticles. I am being a biotechnologist, and my field of expertise are also related to the plant biotechnology. And I have worked in the plant-mediated synthesis of the nanoparticles. And basically, I am working on the formulations of different kinds of nanoparticles. Then why plants are the best contenders? अगर हम green synthesis की भी बात करते हैं तो microbes भी हैं green synthesis में और green synthesis में plant भी हैं. Then why we prefer the plants? So this is the main points. One, it is a simple technique. Second, that is a disadvantage of the microbe, like their maintenance. You have to require a lot of effort and money to isolate the pure cultures and to maintain the pure culture of the microbes. While in case of plant, there is no need to maintain the culture. Plants are available everywhere. Being a agriculture country in Pakistan, almost every plant is present, and you can take it easily, bioavailable. Environmentally eco-friendly है, cost effective है कि just you need the metal precursor salt और plant तो आपको free of cost मिल जाएंगे. Next, single pot reaction है, just you have to mix the metal precursor salt with the plant extract. In single pot, you can synthesize the nanoparticles. Next, moderate temperatures hai, you can synthesize them even at room temperature. You just go for modifications of the temperature or increase or decrease of the temperature if you are not gaining results, or if you are aiming to develop different kinds of shapes or sizes of the nanomaterial. Otherwise, most of the time, nanoparticles are synthesized at the normal room temperature and pressure in case of plant-mediated synthesis of nanoparticles. And the most important, no additional surfactant and capping agents are required. While in case of the chemical methods, you need the extra surfactants or capping agents. And same in the case of the microorganisms mediated. Microorganisms can reduce by using their oxidoreductase enzyme the reduction of the nanoparticles, but they cannot cap them. In microorganisms mediated, you again needed the external capping or surface agent. But in case of the plants, the same phytochemicals which have reduced, they can also cap the nanoparticles after their formulation and making them stable. So this is the main point, that in plant mediated synthesis, you need no any extra capping or surfactant or capping agent. Next, no toxic solvent is required, like all plant extracts are made in water, and water is everywhere, and water is the most कहेंगे जो best solvent है, और that is environmentally friendly है और easily available है, तो all plant extracts can be made in the water. Low toxicity है और biocompatible है. So these are the main points or the reasons that why we prefer plants for synthesis of nanoparticles. Now, what is the chemistry behind plant-mediated synthesis of nanoparticles? Basically, the synthesis of the nanoparticles in plant-mediated case take place in 3 stages or 3 cycles. One can be called as the reduction process. Second is called as the growth phase or growth process. And the third one is called as the stabilization or capping process. In the first step, that is called as the reduction step. Up now, what is the basic chemistry behind that? कि how we think that कि हमने एक salt precursor लिया और उसको एक plant extract से mix किया और just mixing से nanomaterials बन गए. Actually, in what is the chemistry? You are using first initially a metal precursor. I will again give the example of the silver nanoparticles. There are thousands of papers have been published on the synthesis of the nanoparticles by plants. For example, you have taken the silver nitrate as a metal precursor salt of the silver. You have to prepare of any definite molarity stock solution of the silver nitrate, like you can prepare 1 millimolar, you can prepare 2 millimolar. You have to optimize according to your reaction. Let's suppose we have prepared 1 millimolar silver nitrate solution for preparation of the silver nanoparticles, and then you have taken a plant which is rich in phytochemicals, and you have just simply prepare its extract. Let's suppose you have taken 10 grams leaves from the wild plant, and then wash it, and then grind it, and then simply boiled in distilled water, and after boiling you have filtrate it. That is a 10 gram per 100 ml water, the stock solution of the plant extract. Then you have to mix them in a definite molarity, in a definite ratio, like you can take 1 ml of the silver nitrate stock solution, and you can take 1 ml of the plant extract solution. When you mix them with a specific ratio, then what actually happened? Actually, the silver nitrate, when salt is when dissolved in the water, it dissociates into its ion. Silver Ag plus ions plus nitrate NO3 minus 1 ions. These silver ions that are the cations, Ag plus 1 ions, these are unstable, and we have to reduce these ions into the zero valent form. We have to convert Ag plus into Ag0, and the Ag0, that is the zero valent form, is the nanoparticle. So basically the simple chemistry involved is the reduction. You have to reduce the cationic ions into its zero valent form, and the reduction is carried out by the phytochemicals present in the plant extract. These phytochemicals actually no ma'am, there's just inorganic जो natures हैं, like zinc oxide है, silver है, titanium है, they can be, but they are not stable. Like जो zinc है वो ऑक्सीजन के साथ डाइमर बनाती है, तो वो ऑक्सीजन की फॉर्म में ही होता है, वो हम प्योर जिंक नहीं कर सकते. तो नॉट ऑल द मेटल्स, बट सम मोस्ट familiar metals whose nanoparticles have been synthesized. Then after zero valent ion, they are unstable, and or they start to agglomerate with each other. Then again the phytochemicals present in that plant extract, that might be different. ये necessary नहीं है कि जिन phytochemicals ने reduction की है वही phytochemicals जो हैं वो उनकी capping भी करेंगे. Plant extract are a diverse or rich sources of phytochemicals. In single 1 ml of the plant extract there might be 1,000 phytochemicals, 1,000 different nature of the phytochemicals. You can also work on isolated phytochemicals, but we are working as a whole plant extract, but you can also work on the isolation of a single metabolite and can work on that synthesis. So these metabolites can also cap the nanomaterials, which are basically stabilizing them and preventing them from agglomeration. The stability of the nanoparticle is the main factor for their applications. Either you are using in biological applications, or either using for electronics, chemistry, or any other applications, you have to stabilize it. If they are not stable, they will agglomerate and they will lose their unique properties. Therefore, stabilization process is very important. So basically there are 3 stages. One is the reduction, second is the growth phase, and third one is the termination or stabilization or capping phase. And in this figure also we have given the structure of a phytochemical in case of silver nanoparticle synthesis, which are acting as a reducing agent or bioreductant and reducing the silver into zero valent form, which then grow from the silver nanoparticle, and these silver nanoparticles are then again stabilized by these Scandi metabolites. Now, after synthesis, if you have synthesized the nanomaterials, then we cannot use them into any kind of applications superior to their characterization. ठीक है, इसमें ये नहीं है कि आपने नैनो पार्टिकल्स बना लिए हैं, आपने भी जस्ट बनाए हैं. You didn't know about its physiochemical properties. You have to know about all its physiochemical properties. Mainly जिस properties में हम interested होते हैं in biological fields, like size. What is the size of the nanoparticles? What is the shape of the nanoparticles? What is the crystalline nature of the nanoparticles? What is the surface morphology or surface topology of the nanoparticles? What is the surface charge on the nanoparticles? So these are some basic physiochemical properties that we should know before applications of the nanomaterial. And the applications of the nanomaterials are also dependent on these properties. Some properties, on these applications, some properties are size based, applications are size based, like उनमें size matter कर रहा होता है. अगर आपको smaller size के required है और आप smaller size करना चाह रहे हैं तो वो application specific है कि smaller size की चाहिए. और अगर आपको पता ही नहीं है कि जो आपने nano particle produce किया है उनका size क्या है तो it's mean कि you cannot use it, you didn't know about its applications कि आप उनको किस applications में use कर सकते हैं. तो after synthesis the next step in green chemistry of the nanoparticle is their characterization. And for characterization there are different techniques which are used for characterization of the nanomaterials. Firstly, one is the UV visible spectroscopy. In UV visible spectroscopy you basically find out the optical properties of the nanomaterials, optical properties like surface plasmon resonance. You just measure the surface plasmon resonance, and in case of the inorganic metals like silver nano particles, UV visible spectroscopy is the first confirmation technique which confirms you yes the nanoparticle has been synthesized, because every kind of the metal give a specific surface plasmon resonance in a specific range of wavelength. Like in case of silver it give absorbance peak in the range of 400 to 480 nanometer. In case of gold it give peak in the range of 500 to 600 nanometer. In case of zinc oxide nanoparticle, let's suppose it give the peak of surface plasmon resonance in the range of 300 to 380 nanometer. So the first initial confirmation technique which confirms yes the nanoparticles has been synthesized is the UV visible spectroscopy. Next is the transmission or scanning electron microscopy. That is basically microscopic techniques which are used to find out the basically shape of the nanomaterials. हम बेसिकली जो नैनो मटीरियल्स, वैसे तो इन टेक्निक्स की और भी बहुत सी एप्लीकेशंस हैं जो फिजिक्स और केमिस्ट्री में यूज हो रही होती हैं, बट बेसिकली एज अ नैनो टेक्नोलॉजी, अगर नैनो टेक्नोलॉजी से अगर हम नैनो मटीरियल्स की ट्रांसमिशन टेम या सेम करवा रहे हैं, देन वी आर इंटरेस्टिंग टू फाइंड आउट द शेप ऑफ द नैनो पार्टिकल, लाइक आइदर दे आर स्फेरिकल इन शेप, आइदर दे आर रॉड टाइप, आइदर दे आर ट्राइंगुलर, आइदर दे आर octahedral. What shape of the nanomaterial? That is the basic information that we get from the transmission electron microscopy. The second information that we can get from the transmission electron microscopy is the size of the nanoparticles. You can also calculate the size of the nanoparticles by analyzing them either SEM or TEM. But for size, precise calculation of the size, SEM and TEM are not the ideal techniques, but you can have an idea about the sizes of the nanomaterials. So basically the main information in which we interested, that is the shape by which SEM or TEM. If we talk about either SEM is preferred or either TEM is preferred, and if you have a choice between both of these techniques, then TEM is preferred. It has a high resolution, it can generate 3D images, it can generate bright and contrast images, and gives you a clear picture about the shape of the nanomaterials as compared to the SEM. Next is the XR diffraction analysis. XR diffraction analysis is basically used to find out the crystalline nature of the nanomaterials. The basic information you are getting by analyzing this technique is the crystalline nature of the nanomaterials. Either they are face-centered cubic, either they are monoclinic, either they are orthorhombic, either they are hexagonal system. किस class से वो belong करते हैं in their crystalline properties, that is find out by using by analyzing them the XRD technique. The next is the energy dispersive X-rays analysis. Energy dispersive X-rays analysis is inbuilt property of the SEM and TEM. That is always used in conjunction with SEM and also with TEM. In case of nanomaterials, we are interested in the EDX analysis to find out the elemental composition of the nanoparticles. Like we have synthesized silver, then we have to confirm either silver is present in that or not. So this is the elemental composition. Similarly, let's suppose we have synthesized zinc oxide nanoparticles, then the EDX analysis which tells us zinc and oxygen both are present in it, and other than zinc or oxygen, either any other elements are present, and if they are present, then in which ratio, by mass and by atomic number. That all informations are given by the energy dispersive X-rays analysis. Next is the dynamic light scattering analysis. Dynamic light scattering analysis also give about the uniformity of the sizes. Okay, what is the distribution of the nanoparticle sizes? Like might be there is 1,000 number of nanoparticles that have been prepared in your sample, and in from 1000 might be हो सकता है कि जो 100 particles हैं वो 20 nanometer के size के हैं, जो next 300 उसमें nano particles हैं वो हो सकता है कि 80 nanometer के sizes के हैं. To find out the uniformity or particle size distribution, we do the dynamic light scattering analysis. The next is the Fourier transform infrared spectroscopy, abbreviated as FTIR analysis. The FTIR analysis is basically carried out in case of nanomaterials. वैसे तो इसके और भी बहुत से functions हैं जो कि chemistry वाले करते हैं. Either they have synthesized a novel compound, तो वो इसको पूरा analyze करते हैं FTIR spectroscopy से. But as a nanotechnology, if we are going to done the FTIR analysis, then we are particularly interesting to find out which biomolecules are responsible for reduction and capping of nanomaterials. And if you want to know which biochemicals are capping the nanomaterials, then you have to do the FTR analysis, which gives you information about all kinds of bonds and all kinds of atoms that have been capping by your nanomaterials, like carbon-carbon oxygen, carbon-carbon bond, carbon-carbon double bond, carbon-oxygen bond. Each bond जो है that vibrate with a specific set of wave energy that given जो histogram और आप कह लेंगे उसका जो graph होता है उसमें every informations होती हैं जो हम आगे discuss भी करेंगे. Last is the zeta potential analyzer that also gives about surface charge and also the size of the nanoparticles. Now, after characterization, what are the applications of the nanotechnology or nanomaterials you have synthesized either from any of the source, from biological sources, from chemical sources, or either from physical methods. So the main applications, these are in the all areas of life, like nanomaterials are currently being used in agriculture industry. In agriculture industry, if we basically specifically talk about the plant, so there are different kinds of nanomaterials that are currently used for crop movement, like many nanoparticles used as a nano fertilizers, as a nano pesticides, as a nano herbicides, as a nano fungicides, as a nano biosensors, as a nano encapsulations for delivery of the micro and micronutrients to the plant that have overall improved the crop improvement. Similarly, if we talk about the food industry, in food industry, nanotechnology is also being used, and there are so many applications in the food industry. If we specifically talk about food industry, then there are the two main applications, or there are two main spheres in which nanotechnology is being used. One is in food processing and other is in food packaging. In food processing, different kinds of nanomaterials are used as antimicrobial agents during their processing. They protect the food from contamination, from destroying, and some of them are using as anti-caking agents that prevent the foulation or emulsions formations in the frozen foods and other kinds of preserved foods. Similarly, if we talk about the food packaging, then there are nanotechnology has introduced three different kinds of packaging in the food industry. One is called as the active packaging, smart packaging, and intelligent packaging. In active packaging, the packaging of the food is carried out by such material which contain the antimicrobial agents. And in the smart packaging, the nanomaterials are introduced into the food which are actually acting as an indicator either food is safe to use or food has expired. Simply these nanomaterials change the colors, and you can check the expiry date of the nanomaterials by their packaging. Either this food is expired or either this food is fresh to use. So different kinds of packaging have been introduced by the nanotechnology. The next is in electronics or computing. If we specifically talk about electronics or computing, so we all know that कि जो electronic industry है वो बहुत ज़्यादा revolutionized हुई है. Smart watches, digital watches, smart mobile phones, desktop screens, जिसमें different kinds के जो semiconductor materials है, like quantum dots हैं, carbon tubes हैं, flexible display sheets, nano chips for different diagnostic purposes, nano transistors, that all have been synthesizing by using the nanotechnology. Next is the catalysis. Catalysis basically belong to the chemistry. Nanotechnology has also revolutionized the catalysis field. I will like to give the examples of the two major industrial processes of the chemistry. Like one is the synthesis of the ammonia. आई नो कि इतना आप सबको पता होगा, हम बेसिक अपने एफएससी या मेट्रिकुलेशन में पढ़ते हैं सिंथेसिस ऑफ द अमोनिया बाय हाइबर प्रोसेस। और हम क्यों अमोनिया सिंथेसाइज करते हैं कि व्हाई इट्स अ बिग इंडस्ट्री, बिकॉज अमोनिया इज द मेन सोर्स ऑफ फर्टिलाइजर। आप फर्टिलाइजर्स की सिंथेसिस में अमोनिया हर किस्म के जो मेन सोर्स यूज होता है। एंड कन्वेंशनली द अमोनिया सिंथेसाइज बाय यूजिंग द हाइबर प्रोसेसेस यूज डिफरेंट काइंड्स ऑफ convention like titanium, like iron based catalyst, which has a low efficacy, low speed, and also prolonged durations. But now different kinds of nano based catalysts have replaced this industry, which have speeded up the production of the nano ammonia synthesis, and also they are biocompatible or eco-friendly. So they have speed up the synthesis of the ammonia by the Haber process more than 2 times. Similarly, another industrial process that is the hydrogenation of the घी और ऑयल हम बनाते हैं। घी जो है वो सैचुरेटेड है और ऑयल अनसैचुरेटेड है। यस, फॉर्मेशन ऑफ घी जो हम बनाते हैं, तो घी जो है उसमें हम हाइड्रोजनेशन कर रहे होते हैं ऑयल की। तो दैट इज आल्सो अ industrial process in which we also use a nickel as a conventional catalyst, which sometimes recent studies have shown that they are accumulated in our organs. If we are consuming ghee, that nickel accumulates in the ghee. So therefore the nanotechnology has given a replacement of the nickel catalyst with the nano catalyzed, which have speed up the hydrogenation reactions more than the 5 times and with no toxicity or with no toxic effects. Then the automobile industry मैंने already discuss की है, different kind के like buckyball sheet papers, carbon sheet papers have been designed जो कि इतने lighter in weight हैं कि वो flight अगर हम स्पेस इंडस्ट्री की बात करें तो वो फ्लाइट टाइम्स को रिड्यूस कर रहे हैं। लाइक अभी एक रीसेंट स्टडी पब्लिश हुई थी जिसमें कार्बन बकी बॉल्स और कार्बन शीट्स जो हैं, ग्रेफीन शीट्स जो हैं, वो एयरक्राफ्ट इंडस्ट्री में उनकी पेंटिंग की गई, एयरक्राफ्ट की आउटिंग कवरिंग की गई और फिर ट्रैवल जो जेट्स हैं स्पेस क्राफ्ट के वो चेक किया गया कि फ्रॉम अर्थ टू मून जो है वो 2 टाइम्स जो है वो फ्लाइट टाइम रिड्यूस हो गया बाय यूजिंग दीज बकी बॉल्स शीट्स। दे हैव बेसिकली रिड्यूस द फ्रिक्शन ऑफ द एयर एंड आल्सो दे आर वेरी लाइटर इन वेट एज कंपेयर टू द स्टील और अदर conventional kinds of materials. Next, you can use nanotechnology in defense and in military. In defense and military, different kinds of textiles, they have generating the textiles of different colors that can be camouflaged. Like you can electronically gated textiles or dresses which can change their color. You can get it green. If you want to camouflage it, you can change into the skin or dust color. These are the main which can you use in defense. Other than that, IT field, intelligent devices have been devices by using the nanotechnology for defense purposes or for military purposes. Similarly, different kinds of biosensors which can detect the biological weapons, which also increase the defense of any country. Then textile industry is the same as I am discussing different kinds of camouflage clothing. And in a recent study, the scientists of the Harvard University have made a nano sprays. These nano sprays are simply the emulsions of the nano materials with the cotton, conventional cotton, जो कि हम cloth industry में या textile industries में use करते हैं, उनके fibers के साथ उन्होंने nano particles को mix किया है और वो spray just आप बॉडी पे स्प्रे करें तो आफ्टर ड्राइंग दे विल बी कन्वर्टेड इनटू द क्लॉथ दैट विल बी जस्ट लाइक सेम फ्लेक्सिबिलिटी लाइक सेम एंड यू कैन डिसॉल्व इट अगेन। मतलब वो रीन्यूएबल भी हैं, आप उसके बाद उनको सेम सॉल्वेंट में डिसॉल्व करें, देन अगेन स्प्रे इज रेडी, यू कैन डू इट दिस एंड यू कैन कैरी इट विद यू इन योर बैग्स फॉर एनी काइंड्स ऑफ क्लॉथिंग यू कैन डिजाइन। जी। Similarly, in energy generation, nanotechnologies giving a huge bump, like in nano fuels, nano batteries, nano generators, nano transistors. There are different applications in which nanotechnology is playing its positive role. Next is the construction industry. In construction industry, like lighter weight materials have been produced, other than nano paints, nano fillers, which are giving that required strength to the construction agency, and also different kinds of the nano paints, which are dust repellent, water repellent, which are being used in the construction industry. The last one is the main domain, that is the domain of life sciences faculty, that is the health care industry. The next, we are going to discuss the applications of the nanomaterials in the healthcare sector. The nanomaterials, different kinds of nanomaterials have been used and currently in use, like dendrimers, liposome, quantum dot, mesleys, nanospheres, nanoparticles, polymeric nanoparticles, and carbon nanotubes, which has been used for different kinds of biological applications, like for gene therapy, for treating different kinds of genetic disorders. They can, like if we talk about the gene therapy, I am not the molecular biologist, मुझे इतनी details नहीं पता, लेकिन ये applications पता है कि हो क्या रहा है. Basically जो हम conventional vectors use करते हैं for delivery of the genes, sometimes they are degraded by the nucleases enzymes. To protect them from these nucleases degradation, we use the nano material as a nano carriers of the genes. They, one, protect from the degradation, and secondly, acting as for a targeted delivery of the genes by surface functionalization of the nanomaterials. Similarly, different kinds of nanomaterials are used in drug delivery. अगर हम specifically healthcare sector में बात करें तो nanotechnology का जो main या booming field है वो drug delivery है. अब drug delivery में क्या है कि they have revolutionized the world with the word of targeted drug delivery. Nanoparticles can deliver the drug to your target more precisely as compared to the conventional drug systems. Basically, nanomaterials can act as a nano drug and they can also act as a nano carriers. मैं आपको थोड़ा सा briefly इसमें differentiate करूँगी. जो नैनो मटेरियल्स हैं सिर्फ समटाइम्स हम ये मतलब कॉन्सेप्ट ले रहे होते हैं कि जो नैनो मटेरियल्स हैं वो इटसेल्फ as a nano drug काम कर रहे हैं. Yes, they are working, and sometimes they are itself not working as a drug, but just they are acting as a carrier of the drugs. So nanomaterials can act as a nano drugs, and also they can act as a nano carriers. For example, in case we can say if nanoparticles are acting as a nano drug, then what is the example? Cancer, one is the most leading infectious disease causing death और the worldwide. हम सबको पता है कि कैंसर जो है वो एक लीडिंग काज है डेथ की. तो कैंसर की ट्रीटमेंट में नैनो टेक्नोलॉजी का बहुत ज्यादा पॉजिटिव रोल है इन एवरी एस्पेक्ट, आइदर फ्रॉम डायग्नोस्टिक टू द ट्रीटमेंट ऑफ द कैंसर. During the cancer treatment, nanoparticles can be used for the treatment of the cancer, in which the two main conventional therapies have been coupled with the nanotechnology. One is called as the photodynamic therapy and other is called as the photothermal therapy. These are the two main therapies which are used, were conventionally used for the cancer treatment, but now it has been coupled with the nanoparticles, in which different kinds of nanoparticles, like they are called as the photosanitizers. They are sensitive to the light. They have been administered systematically through our systematic system to the person, and जब वो नैनो पार्टिकल्स जो हैं उनको हम सरफेस फंक्शनलाइज करते हैं कि जहाँ पे हमारे कैंसर सेल्स हैं वो बेसिकली हम उनको लिगेंड्स अटैच करते हैं. वो लिगेंड्स एनी काइंड के, वो सरफेस मेम्ब्रेन रिसेप्टर्स हो सकते हैं, वो एंटीबॉडीज हो सकती हैं, वो स्पेसिफिक एप्टामर्स हो सकते हैं, कि वो हमारा स्पेसिफिक जो कैंसर सेल्स हैं वो उन्हीं को जाके टारगेट करें. जब उनमें टारगेट करते हैं और उनमें एक्यूमुलेट करते हैं तो फिर वो जो मेन सोर्स है उसको एक्सटर्नली जब हम लाइट से एक्सपोज करते हैं तो दी स्पेसिफिकली नैनो पार्टिकल्स हैव द प्रॉपर्टी टू अब्सॉर्ब लाइट. जब वो लाइट को अब्सॉर्ब करते हैं तो लाइट को अब्सॉर्ब करने के बाद दे स्टार्ट टू वाइब्रेट, एंड वंस दे आर वाइब्रेटेड दे ट्रांसफर देयर वाइब्रेशनल एनर्जी इनटू द हीट एनर्जी, एंड द कैंसर सेल्स आर वेरी सेंसिटिव टू द हीट, एंड द एरिया ऑफ द कैंसर सेल्स इज हीटेड अप which causes the apoptosis, or you can say the death of the cancer cell. That is coupled with conventional photothermal therapy, in which nanoparticles are acting as a nano drugs. This was the example in which they are acting as a nano drug. Now talk about the nano carriers. Nanoparticles also can act as a can also act for the targeted drug delivery. If we talk about the targeted drug delivery, then you all know that the targeted drug delivery, it is also of two types. One is called as the passive targeting and other type is called as the active targeting. In both kinds of targeting, either it's active targeting or either it's passive targeting, nanoparticles have been used. Like the passive targeting works on the retention time, in which we increase the retention time of the drug in our cancerous cells. And in the active targeting, we are working on the specific targeting to specific cancer cells. इसकी just मैं एक example दूंगी कि active targeting और passive targeting में जो है nanoparticles या nanotechnology किस तरह से help कर रही है. Like जो nano materials हैं और जो nanoparticles हैं, दे पास थ्रू जो सर्कुलेशन सिस्टम, वो आपके सर्कुलेशन सिस्टम से पास होते हैं, आप उनके सर्फेस को फंक्शनलाइज कर सकते हैं. लाइक यू वांट टू टारगेट टू द कैंसर सेल्स, देन यू कैन मेक द एंटीजेंस ऑफ द सर्फेस रिसेप्टर ऑफ द कैंसर सेल, एंड एवरी कैंसर सेल हैज अ स्पेसिफिक सर्फेस रिसेप्टर्स और एंटीबॉडीज. यू कैन फंक्शनलाइज और कोड द नैनो मटेरियल सर्फेसेस विद दैट काइंड ऑफ antibodies, and you can load the drug in the core of the nanoparticles either through incorporation method or either through the adsorption method. And once you are taking either systematically or orally that medicine, so these nano carriers that are carrying the drug will goes towards your target. They will not open up anyone to the cell. They are specialized, surface specialized with the antigens of the cancer cells. So once they will find the cancer cells through the circulating blood circulation, then they will open up, or their matrix is biodegradable, it will be dissolved and it will release the drug to the target. This is the active targeting. Now, what is the passive targeting? In passive targeting, we didn't functionalize the nanomaterial surfaces with the surface receptor of the cancer cells or any other disease, in which just we work on the retention time, specifically in case of cancer. Like we all know that जो cancer cells हैं, they are fast growing cells. और जब वो फास्ट ग्रोइंग सेल्स होते हैं तो जो उनका वस्कुलर सिस्टम है, आर्टरीज और वेन्स, वो पोरली डेवलप्ड होती हैं. दे आर नॉट फुली फंक्शनल बिकॉज दे आर डिवाइडिंग आफ्टर एवरी मिनट. वो डिवाइड हो रहे होते हैं ना, वो इतनी ज्यादा फास्ट उनकी रेप्लिकेशन या मल्टीप्लिकेशन होती है कि उनका जो वस्कुलर सिस्टम होता है वो बहुत पोरली डेवलप्ड होता है. हाँ, पोरली डेवलप्ड होता है. तो उसमें हम भी क्या करते हैं, जो नैनो पार्टिकल्स हैं, जो हम एज ए नैनो कैरियर यूज कर रही हैं, जो कि हमारी ड्रग को लोड किए हुए आइदर इन इट्स कोर और आइदर हमने जो ड्रग है उसकी सरफेस पे एब्जॉर्ब की हुई है, हम उसको फर्दर कैप कर देते हैं विद hydrophilic molecules. Like one of the best example of hydrophilic molecules is the PEG, polyethylene glycol. That is a highly hydrophilic molecules. It can absorb 99% water. वो सेम आप ये समझिए एक पाउडर है, जब आप कोट करते हैं आप अपने वाटर, हम अपने सर्कुलेशन सिस्टम में लेके आते हैं तो हमारी बॉडी में भी वाटर है, तो वो वाटर को अब्सॉर्ब कर लेता है, तो वो नैनो कैरियर जो है वो स्वेल हो जाता है. जब वो स्वेल होता है तो स्वेलिंग से वो हमारा जो सर्कुलेशन सिस्टम है, वेन्स और आर्टरीज से इजीली पास आउट नहीं होता. जो नॉर्मल टिश्यूज हैं वो तो फंक्शनली डेवलप्ड होती हैं, उनमें उसका रिटेंशन टाइम कम होता है, लेकिन जब वो सर्कुलेशन सिस्टम के थ्रू कैंसर सेल्स में आते हैं तो उनका पोरली डेवलप्ड होता है, तो वहां पे समझ लें कि वो स्टक हो जाते हैं. जब वो वहां पे स्टक हो जाते हैं, क्योंकि उनका इतना डायमीटर नहीं होता कि वो स्वेल्ड सब्सटेंसेस को पास आउट कर सके, तो वहां पे नैनो मटेरियल रिजाइड करते हैं फॉर लॉन्गर टाइम एज कंपेयर टू द नॉर्मल सेल्स. एंड इफ दे आर रिजाइडिंग फॉर अ लॉन्गर टाइम इन द कैंसरस सेल्स, देन दे रिलीज द ड्रग देयर, or at least reside for a specific time in which the drug can show their effect. This is a kind of the passive targeting in case of the cancer. So nanoparticles are currently using not only the treating the cancer, for also the delivery of the different central nervous system diseases like Alzheimer diseases, Parkinson diseases, and many other diseases in which it's delivering the drug to their target. हाँ मैम, मुझे नहीं आइडिया इसका. साइबर नाइफ मैंने नहीं सुनी. जी हो सकता है, तो ये मतलब बेसिकली उनके जो साइंटिफिक हो सकता है ये नेम्स हों, लेकिन जो उनके पीछे मैकेनिज्म्स हैं, जो मेन केमिकल थ्योरीज एंड डेट द पीपीटी एंड पीडीडीपी, अब उनके आगे जो मार्केट या लॉन्चिंग नेम्स हैं वो डिफरेंट हो सकते हैं, वो मेरे नॉलेज में नहीं है. Next, they are also using for to treat ocular diseases like हम conventionally जो simple eye rashes है या simple कोई microbial infection है eyes में, तो हम eye drops use कर रहे होते हैं, और उनका conventional disadvantage क्या होता है कि आप eye drop डालते हैं वो निकल जाता है. रिटेंशन टाइम ही नहीं होता, और रिटेंशन टाइम नहीं होता तो वो ड्रग एफिकेसली वर्क भी नहीं कर पाती। तो बेसिकली नैनो टेक्नोलॉजी ने जो है वो रिटेंशन टाइम ऑफ द ड्रग्स पे काम किया है कि इफ यू आर टेकिंग तो दे कैन इंक्रीज द रिटेंशन टाइम, और अगर रिटेंशन टाइम इंक्रीज हो रहा है तो इट्स मीन के फ्रिक्वेंसी ऑफ डोजेस, आप ड्रग की डोजेस भी कम हो रही हैं, एंड दे आर द मोस्ट इफेक्टिव। सिमिलरली फॉर ट्रीटिंग कार्डियोवास्कुलर डिजीजेज लाइक to increase the angiogenesis and to remove the plaque from the arteries and veins, different kinds of nanomaterials have been formulated. Similarly, the stents that are used to treat cardiovascular diseases, the stents have been coated with different kinds of nanomaterials, which are the strongest antimicrobial agents and prevent from infection after post-surgery. So these are the some biomedical applications of the nanomaterials. This was all about what is nanotechnology, what is the main roots of the synthesis of the nanoparticles, and what are their journal applications and what are their biomedical applications. Now, coming towards our specific topic, research-based topic, that is light, an effective stimulator for plant-mediated synthesis of nanoparticles and their biological applications. Just 5 minutes. Okay. So this was the main topic. Actually, मैं उसके बगैर इंट्रोडक्शन के बगैर भी प्रोसीड नहीं कर सकती थी, बिकॉज आई थिंक सो के ऑडियंस इज not from the nanobiotechnology, therefore all should have some little bit information about nanotechnology. So basically this is my research topic. I am working on light. Light is an external effector. Like मैंने अभी डिस्कस किया कि different factors हैं जो कि नैनो पार्टिकल्स की सिंथेसिस को इफेक्ट करते हैं, उसमें से जो एक और इम्पॉर्टेंट फैक्टर है वो लाइट है. आई हैव सीन दैट कि लाइट पे बहुत ज्यादा काम नहीं था. Then I thought कि we should explore the potential of light, different intensities and different wavelengths of light, how they can affect the synthesis of the nanoparticles, and in terms of their controlled synthesis, that how they control the shape of the nanoparticles, how they control the size of the nanoparticles. Then I have done a series of experiment on lights. Short of time की वजह से मैं skip कर रही हूँ, लेकिन मैं अभी brief दूंगी. One was to check the effect of UVC light. This paper is published in International Journal of Molecular Sciences, impact factor 5.9 in 2021. In which we have studied the effect of UVC light. Okay, what is the impact of UVC light on the synthesis of monometallic? One was the silver nanoparticles and other was the zinc oxide nanoparticles, and also on the synthesis of the biometallic nanoparticles, in which the nanoparticles silver by zinc oxide nanoparticles were synthesized in 2 different ratios, and UVC light a strong impact on the shape and sizes of the nanomaterials. Like just to give, मुझे वैसे ये सारे पेपर्स डिटेल में डिस्कस करने थे, लेकिन अब जस्ट गिव टू एन आइडिया अबाउट दीज आर द कैरेक्टराइजेशन टेक्निक्स, XRD to find out the crystalline nature, EDX results to find out the elemental composition of the particles. These are the SEM images of the synthesized nanoparticles. Like in figure 9, A, B, C, and D. A is representing the controlled silver nanoparticles that have been synthesized in dark without any exposure of light. And B represents the synthesis of the nanoparticle under UVC elimination for 2 hours. And you can see a difference in the morphology of the nanoparticles. Nanoparticles, silver nanoparticles, which have been synthesized under the influence or irradiation of the UVC, are more dispersed, no agglomeration, with smaller size and clear spherical shape as compared to the irregular shape of the controlled silver nanoparticle. Similarly, the C and D represents the zinc oxide monometallic nanoparticles. In C, the controlled zinc oxide nanoparticles which have been synthesized in the dark, and in D, it represents the UVC radiated zinc oxide nanoparticles. So UVC has changed the shape of the nanoparticles, and very well defined orthorhombic zinc oxide nanoparticles have been synthesized after the exposure of the UVC light. Similarly, in the case of the biometallic nanoparticles, A represents 0.1 by 0.1 ratio of silver and zinc oxide nanoparticle in control conditions, and B represents the under UVC condition. These ratios didn't show so much promising results as compared when we change the ratio of 0.1 to 0.5, as in case you can see the C, and in the D. In the D figure, you can say there are two different types of material. One is needle type, that is silver, and one is other again orthorhombic type, that is the zinc. So that have clearly differentiated the both types of metals present in the nano materials, in which when we further characterize them after XRD analysis, then they shows the zinc oxide was the most dominated in case of biometallic nanoparticle, and it has masked the effects of the silver. This is the size of the nanomaterials calculated by the XRD data, in which you can see that there is a pronounced effect of UVC on the synthesis of UVC mediated silver nanoparticle was smaller in size than the control, that was 91 diameter, while in the case of the UVC mediated nanoparticles, it was only 22. And these impact was seen in all of the UVC mediated nanoparticles were smaller than their counterpart control nanomaterials. After synthesis of the nanomaterials and after their characterization, these monometallic and biometallic nanoparticles were evaluated for different kinds of biological activities, like antidiabetic activity was evaluated by checking the inhibition of the two enzymes. One was alpha glycosidase inhibition and alpha amylase inhibition, which are involved in the breakdown of the carbohydrates to generation of the glucose. If you inhibit these enzymes, then it means that you can control the production of the glucose in body. So nanoparticles shows very promising results in inhibiting the these both kinds of enzymes, like in figure 11a, alpha amylase and alpha glucosylate. Similarly, the anti-glycation activities was evaluated, in which the advanced glycated end products inhibition was monitored, like vesperin-like and pentosidine. Both types of advanced glycated end products inhibition was monitored by exposing to a specific amount of the nanoparticles, and the UVC mediated nanoparticles shows the highest rate of inhibition, either in case of antidiabetic enzymes and either in the case of anti-glycation enzymes. And at the same time, their biocompatibility or toxicity was also evaluated by performing two assays. One was the percentage hemolysis of the human red blood cells, and other was the LC50 value of the brine shrimp larva assays, and in both cases they show very least toxicity to the red blood cell. That's out of 100, only 3% hemolysis of the human red blood cells was observed. It's mean there were 97% red blood cells were surviving, and nanoparticles are biocompatible. You can use in your in vivo systems for delivery of different kinds of drugs. At the end, anti-cancerous activity of the silver nanoparticles of both types, monometallic and biometallic, was evaluated by evaluating their anti-cancerous activity against the hepatocarcinoma cell lines, HepG2 cell lines, in which we have worked on the 5 factors. Firstly, we have evaluated the viability of the cancer cells. Then we worked the production of the reactive oxygen species and reactive nitrogen species. Then we have also checked the mitochondrial membrane potential of the cancer cells. And then finally, what is the mechanism? How apoptosis of cancer cells is done by the nanoparticles by checking the gene expression of caspase 3/7 gene and also evolving its activity. And in all these results, nanoparticles that were synthesized under UVC light due to their smaller size and unique morphology proven to be the best as compared to the controls. Next, after that experiment, I worked on the effect of UVA and C both. पहले just मैंने UVC का effect evaluate किया. We have also done the experiments with UVB, but there was no result of the UVB, therefore we have skipped, and we published this paper in 2021 in Pharmaceuticals with impact factor 6.3. That is the effect of UV radiation A and C, in which biocompatible zinc oxide nanoparticles were synthesized and their different biomedical applications were evaluated. Due to short of time, I will not discuss the results, just on the same pictures, in which the A and B represent the control zinc oxide nanoparticles, C and D represent under UVA elimination, and E and F represent under UVC elimination. And in this research study, again, the UVC was the most dominating light that can impact the morphology of the nanoparticle in terms of their shapes and also in terms of their sizes. This is the characterization, and this is the anti-cancerous and biocompatibility evaluations of these nanoparticles. And now currently I have completed a startup research program of HEC, in which I have evaluated the effect of LEDs light, in which 5 different LEDs, green, blue, red, yellow, have been checked on the synthesis of the silver nanoparticles. Like this is the different ratios. First, we go through the optimization of the synthesis of the silver nanoparticles, then characterization by UV visible spectrophotometry, then LED mediated. इसमें जो था हमने जो control लिया था वो dark था, without any exposure of light, and then give the 30-minute exposure to different LEDs like green, white, yellow, blue, and red. And the UV visible result shows that कि जब हमने initially we have set the time for 120 minutes, कि हमने 120 minutes के लिए UV light का exposure दिया, sorry, LEDs light का exposure दिया to check the stability कि कब तक वो tolerate करते हैं और वो stable रहते हैं, अपनी morphology को maintain करते हैं. तो in case of the last two one, D and D, that was the blue and red, the peaks become distorted, they become too unstable. Therefore, we have decided the 30 minutes was the approximate time in which all of the LEDs light were stabilizing their nanoparticles. Now there is the FTIR analysis, SEM analysis. Silver nanoparticles of all kinds of LEDs almost shows the spherical nature. एक ही type almost spherical थे. हाँ, there were some refined, like in case of B picture, that was the blue LED mediated silver nanoparticle was amongst the smallest size of nanoparticles synthesized among all LEDs as compared to the others. This is the XRD analysis. This is the size calculation. Next, EDX analysis. Then their biological activities, anti-cancerous activity, biocompatibility, anti-glycation. And next is the summary. So basically, I am working on the light, how different sources of light can affect the morphology of the nanoparticles, and then further these modified or controlled nanoparticles, how can work differentially with the biological applications of the nanomaterial. So as a whole, we can say that nanotechnology is a booming field. It can boost the economy of any country, new therapeutic solutions. It has given new therapeutic solutions to cope diseases and open new frontiers for research in every domain of science. Nanoparticles can be engineered by controlling different parameters in which light prove to be a very good physical factor for trailing of nanoparticles. These are the references. Thank you. Any question? Thank you. Thank you. Yes, sir.
Assalamu alaikum. I'm Jahan Zeeb, serving as a lecturer here in Department of Biotechnology. Thanks for such a wonderful talk and wonderful articles in high impact factor journals.
Thank you.
No doubt, nanobiotechnology is a paradigm shift in area of molecular medicine, but let's talk about some facts. You know, thousands of papers have been published on,
on the toxicity of the nanomaterials.
Nanoparticles, but you see, due to high EPR effect of nanoparticles and due to their size, they get accumulated in tissues.
Yes, in our tissues and organs.
And creating a lot of toxicity and damaging the organs. So what can be the strategy to resolve the toxicity problem and to make them capable for enhanced therapeutic effect?
जी सर, फर्स्ट ऑफ ऑल तो सर मतलब हम जनरली ये बात करते हैं कि कोई भी जो टेक्नोलॉजी है हम उसके कॉन्स की वजह से उसको छोड़ नहीं सकते कि अगर उसके एडवांटेजेस ज्यादा हैं और डिसएडवांटेजेस कम है. लाइक नो डाउट देयर आर थाउजेंड ऑफ पेपर्स हैव बीन पब्लिश्ड ऑन द टॉक्सिसिटी ऑफ द नैनो मटेरियल्स दैट आर दे काजिंग इन विट्रो, लाइक दे कैन accumulate in our body. So the one solution on which the scientists are working is the formulation of biodegradable nanomaterials like lipid nanostructure materials, lipid conjugate nanomaterials, which are biocompatible to human body. They can be degradable, and if they are degradable, they will decompose in the body and will not accumulate or will not cause any potential toxicity to the human bodies. But still, they are causing toxicity to the environment, and hopes okay researchers will find the solutions to cope the toxicity of the nanomaterials.
Okay, thank you. Thank you.
Yes, sir. Thank you, sir. जी सर
using as a lipid
जी
nanoparticles. So
नैनो स्ट्रक्चर लिपिड कैरियर्स हैं, एज अ कैरियर भी यूज हो रहे हैं
जी जी
और एज अ
जी, डॉक्टर साहब।
ड्रग भी
उसमें
यूज हो रहे हैं।
जिस तरह हम पहले पढ़ते हैं कि रिएक्टिव ऑक्सीजन स्पीशीज की अकोमोलेशन हो जाती है, इसी तरह रिएक्टिव नाइट्रोजन स्पीशीज की
जी
है
सर
और
बिल्कुल
लिपिड
ऑक्सीडेटिव
स्पीशीज की
स्ट्रेस काज करते हैं।
जी जी और लिपिड स्पीशीज की भी अकोमोलेशन हो जाती है। तो क्या ये लिपिड्स जो नैनो पार्टिकल्स के थ्रू आ रहे हैं, इट मे आल्सो कॉज द स्ट्रेस ऑन द सेल्स?
जी सर दे कैन काज I have not so much knowledge about this, but I think so they can cause, because these nanoparticles, when we are introducing in the metallic nature, then obviously if they dissolve in the body, they will regenerate the oxidative stress, either in the form of reactive oxygen species or either in the form of reactive nitrogen species. But if they are covered in the biodegradable materials, might be this toxicity will be reduced.
And my second question was that you use the UVC. What was the reason to use the UVC? It is a shorter wavelength and it is a
Yes, sir.
dangerous one. So
सर डेंजरस वन है नो डाउट। बेसिकली जो मेन पर्पस था वो एवेल्युएट करना था कि जैसे यूवीए की जो रेंज है या वेवलेंथ ज्यादा है एज कम्पेयर टू बोथ बी एंड सी, और यूवीसी की जो कम थी। तो बेसिकली माय एम वाज टू एक्सप्लोर आइदर व्हाट लाइट of or what intensity or what frequencies of the lights affects the shapes or sizes of the nanomaterials. I have skipped the mechanisms involved in the light, in which we have discussed the shorter wavelengths being the more energetically absorbs or captured more effectively to the nanoparticles, and the valence electrons of the nanoparticles are excited to the conduction bands, and they affect their surface or optical properties or surface plasmon properties that might affect their shapes and sizes. But what is the accurate, this was the proposed mechanisms that we have proposed, but the accurate mechanism I have didn't find how light accurately or precisely affecting the morphology of the nanomaterials.
बहुत शुक्रिया।
Thank you, sir.
Thank you so much, Dr. Sumera, for such a comprehensive lecture. Ladies and gentlemen, this brings us to end our second talk of the day. Let's take a lunch break and resume the session at 2 p.m. Welcome back. Ladies and gentlemen, we will now resume our second training session of the day. For this session, we have invited Dr. Mariam Javed. She is young and prominent researcher in the field of animal genomics. She is serving as Associate Professor in Institute of Biochemistry and Biotechnology, University of Veterinary and Animal Sciences. Ultimate benefit and sole purpose of her research is to raise the living standards of focal farmers. She has identified various novel genomics regions which provide useful insight into the unique genomic architecture of our indigenous animals. She is contributing substantially to the field of animal genomics in Pakistan. Today, she will deliver the talk on Climb Animal Narrative, a verge of balance between double and deep blue sea. Welcome, Dr. Mariam, on behalf of Virtual University Pakistan. Please proceed.
थोड़े से लोग हैं anyways. Assalamu alaikum. And first of all, thank you very much for giving me this opportunity to stand in front of you and share a few of my work with you. Actually, it's not gonna be a typical lecture or presentation that I have prepared today. It's a kind of interaction with all of you guys. I'll share a bit of what I have done already, and I like to listen from you, what you actually are doing. So maybe together we can develop a collaboration, or we can find the solution of the problems that we'll identify in next few slides. Few years back, I visited Netherlands, and the theme of the year was the climate change and global warming and all that. Before that visit, I never took that topic seriously in my life. And I was like, it's okay, and temperature is fine. We all are doing fine. So what is that hype about the climate change? But once I attended that conference, I came to know that this problem is real and we need to find a solution to that. And since then, I have become an advocate of the problem. And today my topic is quite relevant to that. The topic of my presentation would be Climb Animal Narrative. And we'll try to identify the verge of balance between devil and the deep blue sea. So this figure would show the population rise and challenges we are facing so far for the provision of safe food in developing countries, especially Pakistan. In first figure, you can see that how the population has been increasing day by day. And since like five years or 6 years, in past 5 years or 6 years, you can see that there is a very sharp trend of increasing the population in our region. And in second part, you can see the trend of the population that is increasing in different provinces of the Pakistan. But if you compare the figures that are present on the right side to the figure that is present on the left side, do we have developed the resources to cope up the needs of the increasing population? The answer would be no. Definitely, with increasing the population, there is increase in the pressure in terms of food, in terms of supply of the safe food towards the population to make them healthy. But this is what we are failed to do. And this is the area which we need to focus in our future research. According to an estimate, the demand of the livestock products by 2050 is expected to increasing drastically. If you compare it with the years before, you can see that we don't have much dependency on the animal-based proteins. But with the increasing years, with the passing years, as the population is increasing, we are more dependent on the animal-based proteins. And unfortunately or fortunately, we are part of the society that we love to eat meat. And we are one of the nations in the world which love to eat meat in their, daily food. And it's not only meat, we are also dependent on the animals in terms of other products as well. Like we eat yogurt, milk, egg, and there are many, many products which we are dependent on to get energy. And that is why in recent years, our demand for the animal-based products and animal-based proteins has been drastically increased, and it is going to increase even more in next 25 or 30 years. So it's a big room, it's a big area that we need to develop our strategies in such a way so we can develop the new animal species which can work the way through, and we can develop or we can cope up the need of the population by increasing our production capabilities. This is the world map in terms of total cattle population, because this is the major species which we are focusing for the production of meat. And you can see that US is contributing a lot in terms of 15% of the total cattle meat produced in the region. And then is the Brazil, then is the other European Union and China and Africa and India. And somewhere we are actually existing in the rest of the world portion, which is 13%. Although we are having the best animals in the world, although we are having the best species in the world, best breeds in the world, but still our contribution in the global market is very limited. I think this is because of the lack of strategy, lack of research, lack of genomic exploration of our animal species. That's why we are lacking behind. And this is the area that we need to focus in our future research endeavors. The contribution and importance of livestock sector in Pakistan cannot be denied. We all know that their share in total GDP is much more than many of other countries, like it 53.2%. So livestock is actually contributing in GDP about 53.2%. And the share of livestock in the export of different goods, it's about 8.5%. And the families in Pakistan, which are dependent on livestock in one way or the other, they are 8.5 million families, because Pakistan is an agrarian country, and many of the rural families, they have animals and they actually deal these animals as a expensable commodity. If they are in the need, they will sell their animals and they will get the cash, or they will sell their products and then they will run their families in terms of finances. So they provide raw material for the industry as well, and then social security for rural poor families, and then security against crop failure in Barani areas. But what challenge do we face once we talk about the rearing of livestock? This is to increase the food production, but never compromising the climate that we are affecting actually in the face of climate change. So we need to develop the strategies that we can increase the production of the animals, but not on the face of climate change, which is already affected severely by our various activities. While reducing the carbon cost of the farming or carbon footprints that animals actually are delivering in our environment, but not simply by farming at lower intensity and taking more land because there is not enough. If you travel in the premises of Lahore, you will see that many of the agricultural lands have a board outside and which is written that ye barai furokht hai. It means that we are changing our trend as an agrarian country to the residential. We are changing the land which has been restricted and which should be used for the agrarian activities. And we are now shifting those lands towards the residential plots. And maybe it's real estate is a big business in Pakistan now. And what would happen if we'll keep going like this? Like in 10 years or in 50 years, in coming years, what would happen? That we lost all the lands that we have already designated for the agriculture facilities, and we'll be dependent on the other nations to get even the basics that we are actually growing and we can grow in our country. So challenge faced by livestock. This is what is on the other side. The one thing is on the one side, the benefits that livestock can contribute, but there are different challenges as well, which livestock is also facing contrary. First of all is expanding demand and supply and trading. Okay? Do we have developed our species and breeds so well that they can cope up the need? And feed, food, energy competition, do we have prepared them accordingly? Then livestock diseases, do we have developed the strategies or vaccines or treatment policies to prepare our animals for the future infections? Or safe food supply. Safe food supply is another very big concern because adulteration issue in Pakistan and in the developing countries is very increasing day by day. And last but not the least is climate. Animals are interacting with the climate and climate is affecting to the animals. So both are trying to find their ways to mingle in the middle and to find something that works for both. Impact of livestock on the environment, what it could be. How livestock can impact the surroundings that you live in. What do you think? Do you have any idea that would they affect or impact on the environment? Yes, they do. Actually, they are consuming the food. They are consuming the fodder. That land can be used to grow more of the cash crops, but still you are forced to use that for the livestock. And in terms of methane emission, they are doing a lot. They are doing secretions and their burp, their fart, and all the gases that they are secreting in the environment. They are increasing the temperature day by day, and it will cause the global warming one day. So carbon dioxide, methane, and nitrous oxide, these are three of the main players which are actually contributing negatively towards the environment. And this issue needs to be addressed by research and thorough study. It has been estimated that livestock alone is responsible for 8 to 18% of all the global anthropogenic GHG emission. And you can see in the what happened inside of the animal, that from the behind, from the mouth, everywhere, the methane is actually increasing in the surroundings, especially in those areas where animals are being reared or animals are being formed. So what is the situation of farms which are having animals, and how their environment is actually differing from the environment in surroundings? The water we are getting in that environment where animals have been reared, it's more polluted, its indexes are very high. The animal waste from the factory farms have been polluting actually the underwater resources. The livestock produce a large manure each year, and it's a very big challenge that how we are going to use that and what this can do with your environment. And livestock pollution kills fish and contaminates the drinking water. And this is not only affecting the animals, but as well as the human health. Some big numbers which are quite big concern is that 20 billion domestic animals, they do exist globally. And 45% of Earth's surface actually occupied by livestock system. It means that we are only having 55% of the Earth's surface where we live in. The 33% of the global cropland used for feed production, and that feed is basically consumed by animal. So this is a very big share in total cash crops if you count it. And 8 to 18% of the global greenhouse gas emission is because of the livestock gases. And 72% is the reason of the deforestation. And 30% of the global freshwater consumption has been suppressed or has been compromised because of the pollution that is caused by animals and animal manure. The other side of the balance, as we discussed earlier, that if they are contributing negatively, but still we cannot deny their importance in our environment, that Pakistan is one of the countries who are actually quite reliant upon the protein-based source, which is coming from the animals. If you see in the graph, the maroon line depicts the USA and the very light line depicts the India. Even still, you can see that many of the developed countries, even though they are dependent on the animal-based products and demand of the meat, it's very high in those countries. And that figure shows that contribution of cattle in total meat production is about 37%, and buffaloes are contributing about 21%, and sheep and goat, they are contributing about 26 and 21%. And rest of the meat is coming from other sources. So what are socioeconomic benefits of the livestock? Livestock is significant global asset in terms of value-added products and meat. $1.4 trillion industry hai ye hamare paas. And livestock industry is organized in long market chain that provide income and employ at least 1.3 billion people. So it's a very big number. And livestock GDP, which we have discussed previously as well, it's about 30 to 40%. And in case of Pakistan, it's even more, which is 53.2%. Livestock important as a risk management tool, especially for the poor. And livestock is also key for nutritional security. About 17% of the global kilocalories and 33% of the protein, it is coming from this source. So these are also deliverables in Ethiopia, New Zealand, India, Brazil, Netherlands, China, and United States, where how much protein, actually it is an input-output table, that how much protein we are giving to the livestock and how much we are getting from it. And finally, we have calculated a ratio of both giving an input and output. And you can see that in many of the countries, the output is very high and input is very low, right? It means that they have successfully established a farming system where they are getting more, investing little. So how we can steer a balance of both sides that we can get maximum benefits out of the livestock, but without compromising the environment and climate, which they are affecting negatively. So this is a big question that what we can do in this. It arises over the year is, can we steer a balance between livestock's good and bad? And the answer is yes. And how we can do that? There are several ways. I'm not going to read this out. We'll keep it safe for next few minutes. But few of the methods that I would like to share it with you, it's artificial meat, artificial milk, or international or global market is actually shifting their focus towards these two entities. And then is N-fixing cereals, which are being under trials, and then ruminants producing less methane This we can do by using the advanced genomic methods. So I'll like to give a break here to you all. And we have actually identified the solution in our first half. So I would like you to give me a solution for that. We have identified a problem that livestock is actually how they are contributing and what are different problems that we are facing in Kolekar, and how we can develop the suitable research-based strategies. This is what I want to listen from you. So I would like you all to get a piece of paper and try to give me a research idea which can be used to solve these issues. Can you do that for me? क्या हम ये कर सकते हैं? क्योंकि अभी तक का हमने जो सारा ब्लेम है, if I'll start in Urdu, अभी तक का जो सारा ब्लेम है we put on animals. ठीक है, जैसे इंसानों ने तो कुछ नहीं किया ना, हम कोई गाड़ियां चला रहे हैं जिसमें से धुएं निकल रहे हैं. सो हम कंसिडर करेंगे यहाँ पे इंसानों को स्टैंडर्ड या स्टैटिक, और हम यहाँ पे फैक्टर जो है वो लाइफ स्टॉक को ले रहे हैं, और हम यहाँ पे we'll try to develop a project या we'll try to develop a research based strategy जिससे हम कोई इम्पैक्ट ला सकते हैं और क्लाइमेट को बेहतर कर सकते हैं. तो आई वुड लाइक टू लिसन फ्रॉम ऑल ऑफ यू. You have 5 minutes. Try to develop some idea and then we'll get a discussion on it. Right? आप में से जो कर सकता है वो बता दो ताकि हम डिस्कशन स्टार्ट कर सकें. और if somebody is doing existing research of the project transfer to me as well. क्लाइमेट के ऊपर एनिमल्स किस तरह से अफेक्ट कर रहे हैं, how we can neutralize it. Can we develop a strategy based on research, animal based or non-animal based, to cope up the challenges that we are facing? कोई ऐसी रिसर्च हम कर सकते हैं आपके ख्याल में जिससे हम, I think I'm quite audible. कोई ऐसी रिसर्च है जो हम कर सकते हैं, हेलो, जिससे आप इन चैलेंजेस को cope up कर सकते हैं? If you are already doing a project related to that, तो आप वो भी शेयर करें. तो क्या आपके माइंड में आ सकता है can do better in the department is we have already a research project from microbiology.
Hello, hello, hello. Hello, 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 hello. Hello. Hello, hello.
want to keep audience 5 minutes. I want to keep audience 5 minutes. Then we call one by one. Then we share what we have. Right? Let's take 3 minutes. Start. बाकी कुछ हैं जिन्हें keep up as the other people discuss their issues. Madam, please. Are you working in this area or you want to share some of your experience? No. आपको share करना चाहिए sir, you will not invite me.
Utilize first and neutralize भी as well. अगर आप genetically उनको किसी तरह modify कर सकते हैं कि उनका कोई pathway ऐसा metabolic pathway devise कर लें कि वो methane emission कम हो सके या
Selection of the animals on the basis of this particular
Right.
so we can reduce the emission of the
yes.
The same was I was going to share that. No, I'm from GC Women University, Faisalabad. So I think we can cultivate the bacteria utilizing the methane. Methane can also be used as a biofuel. So I think some interconnected technology must be used so that these waste can act as a substrate for some future technology. Yes, so recycling, recycling. We can put these methane and carbon dioxide into recycling so they can, the other things can get energy as a source of carbon, and then we can increase some biomass and we can make some useful products. The microbes, they are also, we can use them as a protein source. They are the feed additives. So by utilizing the methane, we can grow the methanotrophs, the bacteria which can use this methane, and then that biomass can serve as a energy and carbon source as a feed for the poultry, for the fish, and I think the buffaloes as well.
Excellent.
Thank you.
Thank you so much.
Assalamu alaikum, I'm Dr. Sana. Okay, there could be many genetic solutions to improve the livestock production, but as your topic was related to environment, so I think the main feed that the livestock take comes from the agricultural crop, and pesticides and herbicides are one major factor that may cause toxicity or may cause reduction in the cattle or livestock. So one one remedy could be that instead of using chemical-based pesticide or herbicide, we can use biofertilizers or bio-based material so that they can be biodegradable, less toxic to the animals, and to improve the livestock. Thank you.
I would say that we can use methane as a biofuel, and also we can develop, we can use cell culture to develop artificial meats, like we can culture chicken cells in lab, and we can use that as artificial meat. And I think this has been done in some country in which they have started serving cell culture based chicken in their restaurant. I don't remember that restaurant exactly, but this is this is done there.
Yes, this is happening, but not very proved kind of the methodology. Teacher, please, would you like to share your view on that?
मैम, हम लोगों को वेस्ट मैनेजमेंट सबसे पहले हमें वेस्ट मैनेजमेंट की तरफ भी जाना चाहिए कि जो एनिमल्स की वजह से हम लोग वेस्ट जो हमारे एनवायरनमेंट में है, पूरे जहाँ पे भी फार्म्स हैं या कोई ऐसी जगह जहाँ पर एनिमल्स के साथ डील किया जा रहा है, वहाँ पर वेस्ट मैनेजमेंट भी की जा सकती है। लाइक उसको वो लोग किसी तरह से लेस डेंजरस और यू कैन से लेस टॉक्सिक उसको कर सके बाय यूजिंग सम माइक्रोब्स लाइक नाइट्रोजन फिक्सिंग बैक्टीरिया हो गए या कोई इस तरह की जो कि वो उनके मैनुअल के अंदर मौजूद न्यूट्रिएंट्स को कंज्यूम करके वो उसको डिप्लीट कर सके।
बिकॉज़ बहुत सारे मुल्क इस वक्त इस चीज पर इन्वेस्ट कर रहे हैं कि हम अपने ट्रैश को कहाँ पे निकालें। और few years back there was a petition in the court क्योंकि एक रेजोल्यूशन पास होने जा रहा था for purchasing the trash from Australia और हम अपने डेजर्ट्स के कुछ एरियाज उनको दे रहे थे कि वहाँ पे आप उसको डंप कर दें because they don't want to use their land for that। तो इसके ऊपर एक पिटिशन हुई और बाद में वो चीज विथड्रॉ हुई, बट स्टिल बहुत सारे एडवांस कंट्रीज जो हैं वो शायद उसको अनाउंस किए बगैर they are doing it। What they are doing, their animal-based trash or general trash, whatever they are having, they are transporting that into the developing countries or countries which are deprived, और वो वहाँ पे resources को फैला रहे हैं इस तरह से, which is called diffraction। जैसे डेली पाकिस्तान में आया इस तरह से वेस्ट के साथ और बहुत सारी चीजें भी ट्रांसपोर्ट होती हैं। So it is a very big concern, लेकिन हम उसको किस तरह से रिसाइकिल कर सकते हैं इसके लिए हमें रिसर्च की जरूरत है। वेरी नाइस। आपके साथ जो है शेयर करना चाहेंगे आप लोग? तो पीछे अच्छा, आप सबसे ज्यादा कंसर्न है फिर प्लांट्स होंगे तो
मैम अभी नहीं है ना, अभी हम इनिशियल स्टेज पे हैं, इंशाल्लाह एक टाइम पे आ जाएंगे।
जी आप लोग नो यहाँ से पीछे आप लोग
अस्सलाम वालेकुम मैडम। मैडम एक कॉन्सेप्ट थोड़ा सा कहीं से पढ़ा था फिस्टुलेटेड काउ का, उसमें फिस्टुलेटेड काउज होती है, वो आपको पता होगा बेहतर। लिड लगा के उनके अंदर आर्टिफिशियली फर्मेंटेशन तेज करवाई जाती है। नतीजतन होता ये है कि जो मीथेन प्रोडक्शन है तो वो भी कम हो जाती है, दूसरा ये है कि मिल्क प्रोडक्शन भी बढ़ जाती है। दूसरा जो कॉमनली यूज किया जाता है वो ये है कि जब डंग इकट्ठा हो गया, क्योंकि वो एक बड़ा पैथोजेनिक कंसोर्शिया उसके अंदर मौजूद होता है, तो उसको अगर बायोगैस की तरफ ले जाया जाए तो उससे बाद में जो स्लरी निकलती है वो एज ए फर्टिलाइजर यूज हो जाती है। उसके अंदर पैथोजेनिसिटी खत्म हो गई होती है, क्योंकि बायोगैस प्रोडक्शन के दौरान फर्मेंटेशन होती है, वो सारे का सारा क्लियर हो जाता है। तीसरा ये भी किया जा सकता है कि जो हम चारा खिलाते हैं गाय भैंसों को, हम में से जो लोग गाँव से ताल्लुक रखते हैं या कभी उन्होंने कल्चर देखा हो फार्मिंग का, तो गाय भैंस और ये जो जानवर हैं लाइफ स्टॉक, इसको तूड़ी दी जाती है जो कार्बन से, उसमें कार्बन सोर्स बहुत ज्यादा है। ये अल्टीमेटली काज बनते हैं मीथेन प्रोडक्शन के लिए। तो हम चारे के अंदर भी मॉडिफिकेशन कर सकते हैं, हम वो कंपोनेंट जो अल्टीमेटली काज बनते हैं मीथेन के या उन चीज़ों के जो इन्वायरमेंटल फ्रेंडली नहीं है उसको रिप्लेस किया जाए, यानी जो चारा है इसके अंदर भी मॉडिफिकेशन किया जाए। ये दो तीन बड़े सिंपल से और बुनियादी से आप कह लेंगे
solutions हैं towards this problem, yes, which can
सकते
work for both ends। यानी इससे आप ना सिर्फ एनिमल की प्रोडक्शन बढ़ा सकते हैं बल्कि you can reduce the carbon footprint that actually they are causing। Excellent, thank you। और कोई करना चाहे, or you want to share that you are already doing it?
अस्सलाम वालेकुम, मेरा नाम अब्रार यूसुफ है, बायोलॉजी डिपार्टमेंट से। अच्छा मैम, मैंने एक आर्टिकल पढ़ा था आज से 1-2 साल पहले। एक्चुअली वो था कि उन्होंने जो फिश की गट है उसको यूज़ करके अल्कलाइन प्रोटीज एंजाइम की प्रोडक्शन के लिए, मतलब उसको सबस्ट्रेट एज अ सबस्ट्रेट यूज़ किया था। जो अल्कलाइन प्रोटीज है ये इंडस्ट्रियली काफ़ी इंपॉर्टेंट एंजाइम है।
Yes.
तो इससे सिमिलर स्टडीज़ जो है वो आई थिंक युवास में और शायद पंजाब यूनिवर्सिटी में भी जब वो उन्होंने जो ये चिकन गट को यूज़ करके उन्होंने जो है वो अल्कलाइन प्रोटीज या कोई भी प्रोटीज है वो उसको सिंथेसाइज कराया था। तो इस तरह अगर आप जो कैटल की बात कर रहे हैं, तो अगर हम इस तरह की भी स्टडी डिज़ाइन, आई डोंट नो कि हुई है या शायद ऑप्सो हुई होगी, लेकिन उसको उसके वेस्ट को यूज़ करने एक ये भी तरीका हो सकता है कि उसको हम, क्योंकि वो प्रोटीन, ज़ाहिर बात उसमें प्रोटीन है, मतलब मैं मीथेन के पॉइंट ऑफ व्यू से नहीं बात कर रहा, मैं उसमें जो वैसे अगर हम जो उसकी गट वगैरह अगर फेंक देते हैं जो एक पोल्यूशन ये भी कॉज कर रही है, तो उसको हम एज अ सबस्ट्रेट यूज कर सकते हैं जो इंडस्ट्रियली इंपॉर्टेंट एंजाइम की प्रोडक्शन।
Okay, so your point is that without instead of wasting all the things that we are getting from cow, we need to them or use them in a way that we can get something good from it.
Exactly.
Excellent strategy.
Thank you.
Anybody else? Ji, sure. Is there anybody practically engaged with a project which is dealing with the environment or climate or all that?
Okay, madam, you may definitely you have heard about silage. So it is a fermented type of fodder. So given to the cattle, so it is consumed in lesser quantity, and obviously it will produce less waste. So in Pakistan, it is I think ongoing. My husband itself is undergoing some cattle production.
Marketing तो नहीं कर रही हैं अपनी product?
No, yes. I'm not going to market the product. We haven't made, but we have purchased the silage. So he told me, being microbiology, he shared that it is a fermented fodder. So it is consumed in lesser quantity. You don't need to give the food in tons or in many kilos. So it is just given 1 or 2 times per day, and it gives less waste. I think so. These kind of technologies may help to produce less waste. And if less waste is, then obviously it will be environmental friendly.
Thank you so much. Thank you everybody for sharing your research experience and your thoughts with me. Okay. We have one more.
मैम उसमें अगर आप बायोटेक्नोलॉजी या मोलेकुलर की साइड पर जाएं तो काफ़ी कुछ कर सकते हैं। उसमें जेनेटिक इंजीनियरिंग वाली साइड पर चले जाएं, बैक्टीरिया को मॉडिफाई कर लें, लेकिन मेरे ख्याल से उससे भी पहले हमें थोड़ा सा ये देखना चाहिए कि लाइफ स्टॉक की हमें ज़रूरत क्यों है। तो उसकी ज़रूरत हमें पड़ती है मतलब फूड के सोर्स के तौर पे। तो अगर हम ये करें कि इसकी कंजम्पशन जो है या जो लाइफ स्टॉक पे जो बर्डन है इसकी प्रोडक्शन का, उसको कम करें, तो उससे भी जो है वो कह लें कि जो इससे इसका रोल आ रहा है वो मतलब कम हो जाएगा। मतलब अगर हम मीट और मिल्क जो है वो कम कर दें यूज़ करना, और हम जो है वो वेजिटेरियन साइड पे या वेजिटेबल्स की साइड पे चले जाएं, तो बड़े बेसिक लेवल पे या इनिशियली जो है वो हम उसको काफ़ी हद तक जो है वो कंट्रोल कर सकते हैं। आगे वही है, क्रिस्पर कर लें, जेनेटिक इंजीनियरिंग कर लें, और फर्मेंटेशन को करके भी जो है वो आप उसको कर सकते हैं। लेकिन पहले लोगों को बताना कि आपने उसकी प्रोडक्शन कितनी करनी भी है या नहीं करनी, तो मेरे ख्याल से वो बेटर रहेगा।
अच्छा, ये तो वो बात है ना कि आप खाना ही छोड़ दें।
नहीं मैडम, अगर
दिस इज एक्सीलेंट, और बहुत सारी
उस पे लगा रहे हैं, अगर हम वेजिटेबल्स पे जाएंगे तो मीट की वजह से काफी सारी डिजीज भी आती हैं।
Yes, this transition is actually very hard to induce. In many of the advanced countries, they are working on these trends for being vegetarian and removing the meat totally from your diet, or there are many specific diet plans which are actually encouraging what you are saying, but it's really hard to convince people to do that. But this is again one of the things that we should do. पहले पीछे से एक सुन लेते हैं। पीछे आप कुछ इसमें ऐड करना चाह रहे थे?
जो सोर्सेज है ना वो मौजूद है, मतलब प्रोटीन ही लेते हैं ना अल्टीमेटली, तो बहुत से प्रोटीन सोर्स ही है। तो वो प्रोटीन अगर आप प्लांट से लेना शुरू कर दें, या बहुत सी एल्गी स्पीशीज हैं ऐसी जिनके अंदर बड़ा हाई प्रोटीन कंटेंट मौजूद है, तो आप बड़े आसानी से रिप्लेस कर सकते हैं।
Yes, we already have alternatives which we can go for.
और बड़ा इस्तेमाल हो रहा है। अगर आप यूरोप अमेरिका में, मैं एक छोटी सी मिसाल पुट करता हूँ, एक एल्गी है ब्लू ग्रीन एल्गी स्पाइरुलिका, उसकी आप प्रोटीन कंटेंट अभी जरा चेक करें, वो मोर देन 55% है, आपके ब्रायलर से भी ज्यादा है। और उसको उगाना भी बड़ा आसान है, बीमारियों से भी साफ है, यानी मुर्गी तो आए दिन मर जाती है, उसने तो नहीं होना। उसके और भी, खैर ये तो डिबेटेबल इशू है, मैं उसके बेनिफिट्स या प्रोज एंड कॉन्स बताना शुरू करूँ वो एक लंबी डिबेट हो जाएगी। यानी अल्टरनेट जो है, कहने का मतलब ये है कि वो मौजूद है। हाँ, ये जो आप बात कर रही है ये किसी हद तक काबिल गौर है, इसके ऊपर थोड़ा सोचा जा सकता है कि लोगों को कैसे कन्विंस करना है कि आप इस तरफ आ जाए। लेकिन ये ऐसा इशू नहीं है कि नहीं किया जा सकता, किया जा सकता है। हमें इसलिए लगता है क्योंकि अभी तक स्टार्ट ही नहीं हुआ हमारे यहाँ, वरना लोग इसे टेबलेट फॉर्म में भी यूज कर रहे हैं, लोग इसे डायरेक्ट केक्स बना के इसके खा रहे होते हैं, सब्जी के अंदर इसका इस्तेमाल कर रहे हैं। इंशाल्लाह स्टार्ट लेंगे तो
my comment wasn't कि इसको किया नहीं जा सकता। My comment was it's really hard to convince people to follow the trend that we are trying to create.
मतलब हमारे यहाँ इनिशिएट ही नहीं ना ये चीज,
हमारे यहाँ मेहमान आने पे मुर्गी पकना जरूरी है, शादी पे मुर्गी पकना जरूरी है। सो गोश्त इज वेरी एसेंशियल कंपोनेंट हमारी डाइट का नहीं, हमारे सोशल सेटअप का बहुत एसेंशियल कंपोनेंट है मीट। और डाइट तो पीछे रह जाती है ना कि हम खा क्या रहे हैं, तो वी आर व्हाट वी आर सर्विंग इज व्हाट वी आर। तो थोड़ा सा यस फ्रेम शिफ्ट की जरूरत है, अदरवाइज ऑल्टरनेटिव रिसोर्सेज आर देयर। ये एक मैडम यहाँ पे भी हैं प्लीज।
जो कि मीथेन को यूज करके मतलब मीथेन को एज फूड सोर्स, जैसे हमारे जो नॉर्मल फ्लोरा है हम लाइफ स्टॉक के उसमें अगर इंट्रोड्यूस कराते जो मीथेन को यूज कर सकें एज देयर फूड सोर्स।
ठीक है, एक्सीलेंट। देयर आर डिफरेंट स्ट्रेटजीज एक्चुअली, अगर हम इसको कंपाइल करें। कुछ लोगों ने उसको बिल्कुल इनिशियल लेवल पे कट डाउन करने की बात की, विच इज एक्चुअली इम्प्लीमेंटेबल एंड वी कैन गेट बेनिफिट्स आउट ऑफ इट। कुछ लोग कह रहे हैं कि अगर एमिशन हो रही है तो उसको किस तरह से हम बायोफ्यूल की तरफ लेकर जा सकते हैं, एक और बहुत एक्सीलेंट स्ट्रेटजी है जिसके ऊपर हमें काम करने की इसलिए भी जरूरत है कि इन कमिंग 50 और 25 ईयर्स हम वैसे ही डिप्राइव्ड हो जाएंगे ना। बहुत सारे जो आपके नेचुरल बायोफ्यूल्स हैं या नेचुरल जो आपके सोर्सेज हैं फ्यूल के, वो वैसे ही रिड्यूस हो रहे हैं, और वर्ल्ड जो है इस वक्त भी इट्स ऑन द वर्ज ऑफ द स्कार्सिटी की तरफ जा रहे हैं। और कुछ लोग ये कहते हैं कि हमें उनकी फूड में वी नीड टू इंट्रोड्यूस सर्टेन एलिमेंट्स व्हिच कैन रिड्यूस द एमिशन ऑफ द methane if we conclude the session. So excellent talk. Thank you very much everybody for participating. What I have done actually, it's an indirect approach. Indirect means that in UBAS what we are trying to do is enhancing the genetic potential of our animals so that they would be producing more milk, they would be producing more meat, they would be very friendly towards the environment. And if we look into the subtropical zone of the world in which Pakistan is actually also relying, there are different challenges around the year in terms of extreme temperatures. In these regions, the temperature is very high. There is frequent precipitation. Whenever there is high temperature, precipitation rate would automatically increase. There is high humidity, again, due to the high precipitation. 4 seasons, these are blessings as well, but still, the span of the summer is much larger than the winter. The winters are more of the dry. We don't have much precipitation. Then monsoon, then diverse flora and fauna, and finally populous. These areas are quite populous. The population of animal, the population of human, both is very high. In this area, it means that demand and supply is also very high, and we face a lot of challenge in feeding the mouths that we have here. So extreme weathers cause food scarcity. High humidity provides excellent environment for infection, as the hot and humid environment is very good for the germs to grow, and high population load requires nutritious dietary sources to be explored so that we can feed the high population that we are suffering. But whenever there is a problem, the nature has always blessed you with a solution. You just need to find out. Nature has blessed this region with the kind of animals which are well adapted, which are efficient, which are active, and which are productive. And we can call these animals a survivor. The top of the list is buffalo. I have put this animal on the top because I am working on this animal. But actually, buffaloes are quite unique among all because they have the ability to convert poor quality food roughagages into the excellent products. And in terms of meat, in terms of milk, the production potential of buffalo stands out among all the animals that I have put down. The next is cattle. In Pakistan, we have 16, 17 cattle breeds, which are quite unique. And interestingly, we are having Cholistani cattle here, which is considered to be the ancestor of all the cattle in Asian region and in the region of Central Asia specifically. Then we have sheep breeds, goat breeds, and then is the camel. And in Pakistan, actually, we have flavor of all. If you see USA, if you see Australia, there are few species that are existing there. I mean, camel is in Egypt. Australia and New Zealand are more of the goat and sheep kind of countries. And in US, you will see in Texas especially, you will see the cow, right? But in Pakistan, we are having all of these species and we are getting benefits from this. The camels, they are the animals of the future because they are well adapted to the warmer environment. And finally is the yak. Yak is well adapted in or hilly areas. So river buffalo, which is an animal of choice for my research and animal of choice for this region as well. Why? Because it is highly adaptable to the warmer climate condition and it is carrying the high production potential. And it can consume the poor quality roughages. You don't need to feed the animal with high concentrates or high quality feed, even if it can give you high production in terms of milk yield itself or in terms of the fat percentage that is present in the milk. And it can give you valuable products like milk and meat. So if we'll see the table which is depicting the data of milk composition. And you can see that buffalo milk stands out in terms of calorie count, and in terms of fat percentage, and in terms of calcium and mineral count. And if we'll see the meat composition table, you will see that fat percentage is again very high, but these are good fat because cholesterol is lowest in the buffaloes. And secondly, we are having iron content, which is very high in buffalo meat. So due to the productive potential, this animal is called black gold of Asia. And we are lucky to have about 42 million heads in terms of population of river buffalo or Neeli Ravi buffalo and Kundi, and there are different breeds. And its contribution in total milk produced in the region is about 68%. It means that major milk which is coming into the market is from buffaloes. And in terms of meat, its contribution is about 24%. So now question here is, if the production is good, potential is good, then where does the problem lies, or why these animals are still suffering, or why we cannot get the maximum benefit? The reason is that buffaloes are poor in their reproduction. They are shy breeders, and their reproductive performance or fertility is quite poor in terms of estrous cyclicity, in terms of age of the maturity. Buffaloes are called late maturer because they mature late if you compare it with the cattle, and their estrous cycle is not very regular if you compare it with the cattle. And the animals are repeat breeder in terms of their reproduction and fertility, and their estrous signs, they are not very visible, not very prominent. So these are some of the challenges that farmers are actually facing to get the maximum, to get the next generation from these animals. So if we cannot maintain the chain of the cyclicity in terms of estrous cycle, then it's very hard to maintain a viable buffalo farm. And second main problem is the recurrent infections in the animal. And the main out of them is bovine tuberculosis. This is one of the most widespread infectious disorder in Pakistan. And in recent year, FAO, WHO, and OIE, they have classified Pakistan as one of the nations where tuberculosis is neglected. And it has been estimated that in Pakistani buffaloes, the prevalence of tuberculosis, it's about 33.7. And it means that every 3 out of 10 buffaloes, they are affected by this. And what possible reason can it be? That it's really hard to draw a line. We like to interact with our animals. If we visit the villages, you will see these scenes very frequently. Bachche upar honge, and we like to interact with the animals physically. And that's why if animal is infected, then they are crossing the barrier, and the issue of zoonosis is there, and infection is actually traveling from buffaloes to the human side. So this is the red zone. These are some of the scenes from the slaughterhouse. And what would happen? That animal with tuberculosis, it would come into the slaughterhouse, and people, they will remove the lung, they will cut that part which is affected, and then they will sell the rest of it. And we don't know that what is happening and what type of meat is actually there which we are eating, right? So this is a very big problem, that animals are infected and we are consuming the meat of infected animals. So two main problems. First of all is the poor fertility, and second is the overall poor status in terms of health. But there are good things as well, that our river buffalo is quite unique in its genomic architecture. First of all is the high heterozygosity. Many of the loci that we have studied in University of Veterinary and Animal Sciences so far, their gene structure actually supports the high heterozygosity. Many of the regions are highly polymorphic. Many of the genes are highly polymorphic. They give you actually a barrier for the selection of the superior regions. And then there are certain mutations which are transversions. Then there are certain mutations which are transitions. Both have been reported in buffaloes. Definitely transversions are less, transitions are more. And then we have also identified certain residual truncations in proteins, which are actually affecting the function of the proteins. And then diversity in certain genes is medium to high. And some of the loci, they are monomorphic. It means that they are less informative. And most of the mutations, they are in line with Hardy-Weinberg equilibrium, which gives you an excellent opportunity to put the animal through the selection process to get superior animals through marker-assisted selection. So genomic investigation in US actually comprises of dairy, meat, fertility, and disease resistance traits. And these are different genes that we have explored so far. And in dairy, you can COLR1, CYP11B1, SCD, all of these genes, majority of them are related with the fat percentage in the milk. Majority of them, some of them are actually responsible for the overall yield of the milk. In meat, this is for the increasing the number of cells. For fertility, mainly we are actually targeting the silent estrus behavior. And in disease resistance, these genes have been explored in animals with bovine tuberculosis. So success of buffalo production system is primarily dependent upon the efficient reproduction system. And for this, we need to manipulate the environment as well as the genomics. In environment, it comes nutrition, season, age, week, and disease. And in genomics, what we have, different genes. I have mentioned few of the candidate genes. This is the data of the AVP, which is arginine vasopressin gene, which is a precursor gene important for the production of estradiol. And interestingly, we have identified a simple deletion in one base, and due to that deletion, actually the functional moiety of the gene, which comprises of 9 amino acids, it has been changed. And you can see that last 2 residues have been shifted, and due to that shift, actually the attachment of the protein to its receptor has been reduced, and due to that reduction there is low production of estradiol. And if there is no estradiol, it means that estrus cyclicity would be highly compromised. This is the population genetic analysis of the same gene, AVP. Its paper is still submitted. And total 5 polymorphic sites were being identified, but chi-square value have been depicted that mutation number 2nd, 3rd, and 4th, they are in line with Hardy-Weinberg equilibrium. It means that these mutations are quite fixed in the population. They are showing the trends which are not changing generations after generations. It means that they are good marker for the association analysis. And this is their association analysis, and the p-value, which is the score of significance of these polymorphic sites. It shows that polymorphic site, which is showing the deletion, which I mentioned earlier, it is actually very high. It means that it is impacting the trait in a very big way. So we have identified 3 genotypes: the wild type genotype, then we have mutant form, and then we have heterozygous form. And you can see that score of heat, it is quite different in all 3 genotypes. So it gives us a selection marker where we can choose the animals with the better trait. This is the 3-dimensional protein structure analysis actually, and it is indicating the structure of the protein. And you can see that secondary structure elements are quite different, like alpha helix and beta strands. And there is an absence of the TM helix in the mutant form. So that helix is actually responsible for the binding of the protein. And due to the change in those 2 amino acids, which we saw previously, the attachment of the protein with the receptor is hindered. And due to that, there is no production of estradiol. That is why animal is suffering from silent estrus behavior. This is the analysis of comparative genomics, in which the river buffalo has been compared with swamp and Mediterranean. And you can see that there are certain regions which are very unique. At 2 places, you can see the deletion in totally in swamp and Mediterranean. But in Murrah, which is quite near to the Neeli Ravi buffalo of Pakistan, and Murrah is from India, but they are having very unique region there. This is the analysis of phylogenetic relationships, exon distribution in gene and motif patterns. And you can see that in cattle, there are patterns which are different, and the same pattern has been followed by swamp and Mediterranean. But in terms of Murrah buffalo, you can see that pattern is totally different, and conserved domain region, and especially C motif pattern at C position, the motif pattern is changed. It means that the protein is highly diverse and protein is changed if we talk about Murrah. So this is the truncation of the protein, right? But it includes the 9 amino acid moiety, and there is a variation in that as well. And this is BB genotype in buffaloes. The rest of the animal species, they are following the same pattern as in the standard. The second work we have done in the toll-like receptor 1, this gene actually was being explored for bovine tuberculosis and its immunity, natural immunity in the animal. And we identified total 5 SNPs even in this gene. And there is also a stop codon. Isoleucine was converting into a stop codon. And due to that stop codon, there was again truncation of the protein, which was being observed in the cattle. It is the genes that we have selected so far. It is quite common that you find some of the regions get deleted, and then, or maybe they are converted into the stop codons. So unique genomic variations have also been identified in interferon gamma gene. In this gene, we have identified total these SNPs. And you can see that proteins are also being affected functionally due to the presence of these proteins. This is the association analysis. And this analysis indicates majority of the regions, they are obeying the Hardy-Weinberg equilibrium. And it means that association analysis shows that they are actually affecting the basic immunity of the animal. So if we select these markers to select the animals with superior immunity trait, it means that animal would survive the infection in upcoming life. CYP11B1 gene, this gene actually is associated with the milk fat content. The fat content actually in milk is measured in two ways. You can calculate the total fat percentage in the milk, or you can actually calculate the total fat in kilograms as well. The CYP11B1 gene was actually studied in Nili Ravi buffalo for the fat overall. And there are different SNPs which are being identified. And due to these SNPs, you can see that there are different genotypes, wild, mutant, and heterozygous genotypes. And this provides us the opportunity that we can select the best out of it. And if you see the scores, the polymorphism, this one, this is highly significant. And the fat percentage is up to 7.9%. It means that if you select the mutant form of this particular loci, the animal can give you very high fat percentage. OLR1 gene is also responsible for the fat content in the milk. These are different variations. Some are transitions, some are transversions, which are being identified in the gene. And here also you can see the first one, and again, the first one and the second last one. These are the two SNPs, which are actually showing great association towards the trait. Then is the SCD. This gene is also responsible for the milk fat content. These are all actually production trait genes. In this also novel polymorphic sites were being identified in river buffalo. And you can see that association analysis provides us with the high values in which we can identify those loci which are showing more of the fat content, and then we can select those animals for future breeding program. The same polymorphic sites were actually put through the three-dimensional protein configuration, in which the protein structure was in silico, constructed in silico, and you can identify certain transmembrane helix, then you can identify secondary structural elements in the protein, and you can see the sites, or you can actually identify the sites of your mutation, that how functionally they are interacting in the biological environment. In OLR1 gene, we have identified two domains. One is steroid binding domain, and second one is the these are two different motifs and two different domains, and you can see that your mutation is also actually existing in those domains. It means that it is affecting the binding sites of the protein. This is the SCD gene result, and you can see that your mutation actually is existing in the transmembrane helix region. So if we conclude these results, being most diverse among the livestock species, especially buffaloes, river buffaloes need extraordinary attention in terms of research and policy to get the maximum benefit out of this astounding and phenomenal animal. And you can see the diversification of this animal and its existence globally. Green color depicts the presence of river buffalo in different regions. The yellow color depicts the swamp buffalo, which is more in China. And where it is orange color, it means that both type of buffaloes are existing in those regions. So this is some of the basic research that we have done so far, in which we have identified certain candidate genes which are responsible for controlling different traits of economical importance in livestock. And if we select the animals on the basis of these traits, it means that we can make these animals better, and these animals would produce better, and these animals would survive in our environment in better way. So what comes next is to use this knowledge to design more of the advanced projects that we actually are doing in US for the selection of those animals, or for making those genes viable which are otherwise non-functional in buffaloes. So we have planned certain projects on the stem cells, then microRNA, then we are also working on the genome editing, and this is a lipofectamine that we are going to make a complex, and then we are going to use it to treat the animal. One of the research project that is in the progress, it is with the collaboration of Royal Veterinary College and the Francis Crick Institute, UK, and student is also here. It is sponsored by Commonwealth, and our title is Study of the Role of CATA3 gene in Cell Lineage and Differentiation, and we would be able to silence those genes which are responsible in the poor fertility of the animal by using CRISPR-Cas9 system. The second research project is about the variant transcriptome analysis in Neeli Ravi buffalo breed. This is actually funded by ALP, which is Agricultural Linkage Program by Pakistan Agriculture Research Council Islamabad. And they are also supporting this project, and many of the students have completed their thesis in it. One of the PhD student is still here who is working on this. And the last one is study of circulatory microRNA as a novel remedial approach for the issue of silent estrus behavior in Neeli Ravi buffalo. This is also a funded project in which what we are going to do is that we are going to isolate the circulatory microRNA, and then we are going to make a complex of these RNA with lipofectamine, and then we are going to use that complex for the treatment of the animal. And that microRNA would actually affect the expression of the gene, and we'll get the expression of our own will. And finally, the production and the fertility status of the animal would be enhanced. So today's activity is actually related to this project. What we are going to do in our next session is that we are going to use that database, that how you are going to select your microRNA. If you are interested in a project like that, there are different databases, just like genes, where you can select the sequence of the gene. Just like that, you can use different databases for the selection of relevant microRNA as well. So my student is going to demonstrate you in next 15 years. So I hope you will cop up with her. Ajain, please, Ikra. If you have any questions so far, you can ask, or we can have the question answer session in the end. Ji sir, please.
Thank you very much. I will start from southern part of Punjab. I joined the lecture a little bit late, but thank you very much. I am more interested towards that slide in which you are showing us that the fat percentage was 7%.
Ji sir.
So when we were student and we were reading, I'm student of agricultural economics. So we know that the cow has 3 to 5% fat percentage, and buffalo is plus minus 5 to 6. So I was really happy to see the result of the 7% fat in the Neeli Ravi buffalo. And most probably at your Ravi campus, you might have that buffalo available with you, and you are in the process of fertility, and you are making as I was just listening there, cloning and other things. So I wish this 7% fat percentage buffaloes genes, and they're through embryo transfer technology or whatsoever the technology you know better, because you are the expert on it, so that our farmers can get the buffaloes having good quantity of fat in that milk. Because in our villages still we love to have a thick layer of
cream on the milk.
Cream
Yes, sir.
on the milk. So we wish, and you are the only university, leading university of the Pakistan on livestock side. So we wish you people should give us a gift to the Pakistan with having this 7% fat buffalo.
Yes, sir.
Thank you.
Thank you very much, sir. We are doing what we can actually. And fat percentage is quite desirable trait. You are right in Pakistan, because in some of the nations, people are quite concerned over the fat content and they are health conscious and they are moving towards the skim milk, but not in Pakistan. So yes, it's not just one animal actually. Once we collected the samples, we collected more of like 150 or even more samples. And then we got some ratio, although that ratio was very low. There were very few animals which were touching the 7% or 7.5%. Majority of them, they are like 5, 5.2, 5.1, you're right. But thank you very much, sir, for your comments. Thank you so much. So now we have Ikra. She is going to demonstrate you about the use of the microRNA software, that how you can collect the data from that.
Thank you, ma'am. Yes, back.
इसको बंद करके आप इंटरनेट का सोर्स ओपन कर दीजिए. ओके, यहाँ से
Assalamu alaikum. My name is Zikra Kosa, and I am a PhD scholar in the field of molecular biology and biotechnology under the supervision of Dr. Mariam Javed at University of Veterinary and Animal Sciences, Lahore. The purpose of my presence here is to tell you about the software which we can use for the microRNA analysis. Actually, there is a long list of microRNA software for the microRNA analysis available online. They are freely available. If I name some of them, such as MIRBase, MIRPath, PICTAR, TargetScan, MIRWalk, and many more. Today, I will tell you about some of them. So let's start with the MIRBase software. It is freely available online, so there is no issue regarding its access. I will write the name in the search bar. Okay. Open it. This is home page for the MIRBase database. It's a biological database, and it contains information about more than 40,000 non-published microRNA sequences in various species. If we talk about some of its main features, is that MIRBase database, it provides a way we can retrieve the sequence of mature microRNA as well as the stem loop precursor, which is its premature form. चूंकि हमें पता है कि जो microRNA है, इसकी एक mature form होती है और इसकी जो premature form है, वो stem loop या hairpin precursor में होती है. तो ये जो सॉफ्टवेयर है MIRBase, ये आपको एक अपॉर्चुनिटी प्रोवाइड करता है कि आप इसमें जो इसकी प्रीमेच्योर फॉर्म है उसके सीक्वेंस को भी रिट्रीव कर सकते हैं, एंड यू कैन आल्सो रिट्रीव द मैच्योर सीक्वेंस. और इसको रिट्रीव करना भी बहुत सिंपल है. जस्ट यू हैव टू मेंशन the name of microRNA in the search bar. You can also use keyword or any other reference. If we talk about some other features of this this MIRBase database, is that it provides a consistent naming system for the microRNA और इसमें इसकी जो naming system है is sequential. इसका मतलब ये है कि अगर हम माउस स्पीशीज की बात करते हैं, अगर उसमें एक लास्ट पब्लिश माइक्रोआरएनए जो है उसका नाम 2 to 4 है, तो जो नेक्स्ट पब्लिश माइक्रोआरएनए होगा उसको 2 to 5 कह दिया जाएगा. तो इसकी जो सीक्वेंस जो इसकी नेमिंग है वो इस तरह से की जाती है. और अगर आपने फर्दर इसमें इंफॉर्मेशन चाहते हैं, यू कैन विजिट द हेल्प पेजेस फॉर मोर इंफॉर्मेशन अबाउट द नेमिंग सर्विस. Other than these features, this MIRBase database, it also provides experimental evidence. Experimental evidence means that it provides an open access to the papers in which this microRNA is reported. So it's a way कि आप उसको verify कर सकते हो, अपने microRNA को वहाँ से verify कर सकते हो. उन papers तक आपको open access दी जाती है जिनमें ये microRNA उस species में report किया जाता है. इसके अलावा अगर हम इसके एक और फीचर की बात करें, प्रोवाइड्स लिंक टू द अदर डेटाबेसेस, जिन डेटाबेसेस में ये माइक्रोआरएनए रिपोर्टेड होगा. तो इस तरह से आपको मतलब इस सॉफ्टवेयर के थ्रू आप दूसरे डेटाबेसेस पे भी जा सकते हैं और आप अपने रिजल्ट को वहाँ पे अपने सीक्वेंस को वेरीफाई कर सकते हैं. Okay, this database is managed by University of Manchester. और हम यहाँ पे अब देखते हैं कि हम किस तरह से how we can retrieve the sequence by using this database. Simply, I have selected microRNA 132. तो मैं यहाँ पे उसका name mention लिखती हूँ, ठीक है? MIR 132, ठीक है? Okay, the next page will be open. और अगर आप देखें तो यहाँ पे वो मैं बता रहा है कि your query microRNA 132 returned with 211 results. तो हमें इतनी सारी various species में इस जो ये microRNA reported है उसका result हमारे सामने आ जाएगा. If I scroll down, आप देख सकते हैं कि कितनी लंबी लिस्ट है और यहाँ से आप फर्दर इसको देख सकते हैं जो भी आपकी इंटरेस्टेड स्पीशी है जिसमें आप इस माइक्रोआरएनए को देखना चाहते हैं. आई हैव सिलेक्टेड MMU. अच्छा, यहाँ पे जो ये फर्स्ट 3 लेटर्स हैं जैसे DRE है, PTR है, दिस फर्स्ट 3 लेटर रिप्रेजेंट द स्पीशी नेम. DRE which is Dino radio which is a zebra fish, and MMU mouse musculus. I will select this. ओके, एज आई ऑलरेडी मेंशन कि ये सॉफ्टवेयर आपको आप इसके थ्रू अपना स्टेम लूप सीक्वेंस भी रिट्रीव करना चाहते हैं, अगर आप उसमें इंटरेस्टेड हैं, या आप इसमें अपना मैच्योर सीक्वेंस भी रिट्रीव कर सकते हैं, ठीक है। एक्चुअली दिस पेज इट कंटेन्स 3 इन्फॉर्मेशन, ये आपको स्टेम लूप सीक्वेंस भी बता रहा है, ये आपको मैच्योर सीक्वेंस भी बता रहा है, और इसके अलावा जो हमने रेफरेंसेस की बात की थी कि इट प्रोवाइड्स एक्सपेरिमेंटल एविडेंस सिस्टम, तो वो रेफरेंसेस जो हैं वो आपको यहाँ पे ये बता रहे हैं, ठीक है। अगर हम वापस इसके ऊपर जाएं और देखेंगे व्हाट काइंड ऑफ द इंफॉर्मेशन वी कैन ऑब्टेन फ्रॉम हियर, सो बेसिक इन्फॉर्मेशन है, उसका एक्सेशन नंबर है, उसका सिंबल, डिस्क्रिप्शन, जीन फैमिली, लिटरेचर सर्च 178 ओपन, इस माइक्रो आरएनए के लिए 178 ओपन एक्सेस पेपर्स हैं जिनके अंदर ये माइक्रो आरएनए रिपोर्टेड है इन द माउस स्पीशी। अगर यहाँ से इसे क्लिक करेंगे तो वो पेपर्स तक आपकी एक्सेस हो जाएगी। ठीक है, तो ये पेपर्स हैं और आप एक इट्स प्रोवाइड, आप कह सकते हैं कि आपको एक वे प्रोवाइड करता है कि आप वेरीफाई भी कर सकते हैं। ओके, डीप सीक्वेंसिंग इंफॉर्मेशन इज आल्सो अवेलेबल हियर। कॉन्फिडेंस, अगर आप यहाँ पे देखें तो लिखा है कि इसका कॉन्फिडेंस लेवल हाई है। इसका कॉन्फिडेंस लेवल हाई होने का मतलब ये है कि यू कैन रिलाई ऑन द ऑथेंटिसिटी ऑफ दिस माइक्रो आरएनए। तो जो अगर यहाँ पे इसका जो कॉन्फिडेंस लेवल हाई है तो आप उसकी ऑथेंटिसिटी के ऊपर रिलाई कर सकते हैं। जीनोम कॉन्टेक्स्ट, अगर आप जीनोम कॉन्टेक्स्ट के पास जाते हैं, इसमें जाते हैं, तो यहाँ पे आपको जो जीन रिलेटेड इन्फॉर्मेशन इट इज लोकेटेड ऑन क्रोमोसोम नंबर 11, इट्स स्टार्टिंग पोजीशन, एंडिंग पोजीशन ऑन द पॉजिटिव स्ट्रैंड, इस तरह की जो इन्फॉर्मेशन है वो आपको यहाँ से मिल सकती है। फिर हमारे पास आता है क्लस्टर्ड मिरनास। जो क्लस्टर्ड मिरनास है, क्लस्टर का मतलब ये है कि सेट ऑफ 2 और मोर माइक्रो आरएनए दैट आर ट्रांसक्राइब्ड बाय द माइक्रो आरएनए जीन्स दैट आर फिजिकली एडजेसेंट टू ईच अदर। मतलब वो जीन्स जो कि फिजिकली एडजेसेंट होंगी एक दूसरे के, जो कि फिजिकली एडजेसेंट हैं, तो उनसे जो माइक्रो आरएनए बनेंगे उनको वो एक क्लस्टर फॉर्म के अंदर होते हैं। जैसे हमारा जो टारगेटेड माइक्रो आरएनए विच इज 132 इन द माउस स्पीशी, ये क्लस्टर है 212। इसका मतलब ये है कि ये दोनों माइक्रो आरएनए की जो जीन्स हैं दे आर फिजिकली एडजेसेंट टू ईच अदर, और इस वजह से ये एक क्लस्टर बना रही हैं। फिर इनकी फर्दर इन्फॉर्मेशन दे आर लोकेटेड ऑन क्रोमोसोम नंबर 11 और पॉजिटिव स्ट्रैंड, ऑन द पॉजिटिव स्ट्रैंड, और इनकी स्टार्टिंग पोजीशन और इनकी एंडिंग पोजीशन की सारी इन्फॉर्मेशन यहाँ पे अवेलेबल है। ओके, एज आई ऑलरेडी मेंशन, इट प्रोवाइड्स डेटाबेस लिंक्स जिनके थ्रू आप दूसरे डेटाबेसेस में भी जाके इस सीक्वेंस को देख सकते हैं। अगर यहाँ पे देखें तो यहाँ पे हमारे पास इंटरेस्ट जीन है, अगर मैं इसके ऊपर क्लिक करती हूँ तो ये हमें लेके जाएगा NCBI पेज के ऊपर, और NCBI पे जिस तरह से हमें इंफॉर्मेशन, एक फॉर्मेट है इंफॉर्मेशन मिलने का, वो सारी इंफॉर्मेशन हमें यहाँ से मिल सकती है। ठीक है, ऑफिशियल सिंबल, फुल नेम, प्राइमरी सोर्स क्या है, और ऑर्गेनिज्म कौन सा है, लीनिएज, अगर इसके सिनोनिम्स हैं वो मेंशन है, समरी। ठीक है, और हम यहाँ से सिर्फ DNA सीक्वेंस को नहीं रिट्रीव कर सकते, बल्कि हम यहाँ से माइक्रो आरएनए सीक्वेंस को भी रिट्रीव कर सकते हैं। एंड वी कैन आल्सो रिट्रीव द सीक्वेंस इन द फास्टर फॉर्मेट फ्रॉम दिस सॉफ्टवेयर। ठीक है, तो हम यहाँ से इसे इजीली रिट्रीव कर सकते हैं। ओके, यहाँ पे अगर हम ये कहें कि इस सॉफ्टवेयर में जितने भी माइक्रो आरएनए जो कि यहाँ पे रिपोर्टेड हैं, पब्लिश्ड हैं, वो तमाम हमें NCBI के ऊपर भी मिल जाएंगे, तो ऐसा नहीं है। सिर्फ वही माइक्रो आरएनए जो NCBI के ऊपर रिपोर्ट होगा उसका यहाँ पे रेफरेंस दिया जाएगा। मेरा कहने का मतलब ये है कि अगर मैं कोई और ऐसा माइक्रो आरएनए लेती हूँ, दूसरा माइक्रो आरएनए लेती हूँ, तो ये ज़रूरी नहीं कि उन सब के लिए भी मुझे यही वाले सॉफ्टवेयर यहाँ पे डेटाबेस लिंक्स में दिए जाएंगे। जिस सॉफ्टवेयर का दूसरे डेटाबेस के साथ लिंक होगा, सिर्फ वही सॉफ्टवेयर यहाँ पे प्रोवाइड किया जा सकेगा। मतलब आप हर माइक्रो आरएनए को NCBI से, अगर उसके ऊपर वहाँ पे सीक्वेंस नहीं है, तो आप उसे रिट्रीव नहीं कर सकते NCBI से। सिमिलरली ये अदर डेटाबेसेस हैं जहाँ से आप इनको जाके इनके सीक्वेंस को रिट्रीव कर सकते हैं। जो स्टेम लूप स्ट्रक्चर है, विच इज जो कि इसकी प्री मैच्योर फॉर्म है, उसके बाद हमारे पास सीक्वेंस है मैच्योर फॉर्म का, मैच्योर सीक्वेंस MIR 1325P एंड 3P। और यहाँ पे अगर हम नोटिस करें एक बात कि जो माइक्रो आरएनए जब एक जहाँ पे भी स्मॉल r लिखा होगा, तो ये जो स्मॉल r है ये रिप्रेजेंट करेगा इसकी प्री मैच्योर फॉर्म को। ठीक है, जब माइक्रो आरएनए MIR लिखा होगा स्मॉल r के साथ, तो इट इंडिकेट्स इट्स प्री मैच्योर फॉर्म। लेकिन जब हमें कैपिटल R के साथ नजर आएगा तो इट्स रिप्रेजेंट इट्स मैच्योर फॉर्म। और हमें यहाँ पे इसकी जो मैच्योर फॉर्म है, यहाँ पे 2 आर्म्स के ऊपर रिजल्ट को दिखाया गया है, 5p and 3p. 5p means the sequence is retrieved from the 5' arm of the stem loop structure, and 3p means the sequence is retrieved from the 3' arm of the stem loop sequence. और मतलब हर माइक्रो आरएनए के लिए ये दोनों सीक्वेंस हमें यहाँ पे नहीं दिखाए जाते। यहाँ पे दोनों जो 5p आर्म और 3p आर्म जो यहाँ पे शो किए गए हैं, इसका मकसद ये है क्योंकि इन दोनों आर्म्स में से जो इनकी प्रीडोमिनेंट स्टेट है वो डिटर्मिन्ड नहीं है। अगर किसी माइक्रो आरएनए के लिए उसकी जो उसका कौन सा आर्म प्रीडोमिनेट कर रहा है, अगर वो फॉर्म डिटर्मिन्ड है तो सिर्फ उस ही फॉर्म को यहाँ पे दिखाया जाता है। लेकिन इस माइक्रो आरएनए के लिए 5p और 3p में से कौन सी स्टेट जो है वो प्रीडोमिनेट कर रही है या कौन सी जो है वो अबंडेंस है वो नहीं पता, तो इस वजह से यहाँ पे ये दोनों सीक्वेंस हमें यहाँ पे दिखाए गए हैं। और इसकी जो इन्फॉर्मेशन है जिस तरह से हमने स्टेम लूप स्ट्रक्चर में देखी बिल्कुल उसी तरीके से ये एक्सेशन नंबर है। प्रीवियस आईडी अगर कोई पहले आईडी थी और उसको फिर रिप्लेस किया गया न्यू आईडी के साथ, अगर किसी माइक्रो आरएनए के लिए है तो वो यहाँ पे शो की जाएगी। सीक्वेंस के लिए है कि सिंपल हम क्लिक करेंगे ऑन द गेट सीक्वेंस ऑप्शन और हमारे पास जो है सीक्वेंस हम यहाँ से इजिली रिट्रीव कर सकते हैं। Okay, deep sequencing information is also available here. Evidence, जैसे मैंने पहले बात की कि यहाँ पे आपको एक्सपेरिमेंटल एविडेंस प्रोवाइड किया जाता है, आप अपने रिजल्ट को वेरीफाई कर सकते हैं। यहाँ पे जो बेसिकली एविडेंस के जो इंफॉर्मेशन है ये 2 सोर्सेज से आती है। फर्स्ट वन इज एक्सपेरिमेंटल एविडेंस, जो एक्सपेरिमेंटल एविडेंस होता है जैसे पब्लिश पेपर्स हैं, और सेकंड होता है कि यहाँ पे जो प्रिडिक्टेड होमोलॉग्स होते हैं फॉर द माइक्रो आरएनए, और जो होमोलॉग्स हैं दे आर प्रिडिक्टेड ऑन द बेसिस ऑफ द सीक्वेंस सिमिलरिटी, फोल्डिंग प्रॉपर्टीज ऑफ द स्टेम लूप स्ट्रक्चर। तो इस बेसिस के ऊपर यहाँ पे जो एविडेंस है वो प्रोवाइड किया जाता है। और अगर फॉर एग्जांपल कोई प्रिडिक्टेड होमोलॉग है और उसको एक्सपेरिमेंट वेरीफाई नहीं कर रहा तो वो इन्फॉर्मेशन यहाँ पे नहीं दी जाती। सिर्फ वही प्रिडिक्टेड होमोलॉग्स में से भी सिर्फ वही इन्फॉर्मेशन आगे आती है जिसको एक्सपेरिमेंटली वेरीफाइड होती है। और डेटाबेस लिंक्स, यहाँ पे दूसरे डेटाबेसेस के साथ भी जो है वो लिंक्स प्रोवाइड किए जाते हैं। प्रिडिक्टिव टारगेट, ये जो डेटाबेस है ये सिर्फ इसके थ्रू हम सिर्फ सीक्वेंस को रिट्रीव नहीं कर सकते, हम यहाँ पे टारगेट को प्रेडिक्ट कर सकते हैं, क्योंकि हमें पता है कि माइक्रो आरएनए हैज टू बाइंड इट्स टारगेट मैसेंजर आरएनए इन ऑर्डर टू डाउन रेगुलेट इट। तो उसके हमें कैसे पता चलेगा कि ये माइक्रो आरएनए है और ये किस जीन में इसका टारगेट है, किस जीन के साथ जाके ये बाइंड करेगा और उसको डाउन रेगुलेट करेगा। तो वो इन्फॉर्मेशन वी कैन ऑब्टेन फ्रॉम दिस सॉफ्टवेयर, क्योंकि ये हमें टारगेट प्रिडिक्ट करके देखता है। एक्चुअली ये जो सॉफ्टवेयर है ये हमें दूसरे डेटाबेसेस के साथ लिंक प्रोवाइड करता है। अगर यहाँ पे प्रिडिक्टेड टारगेट के लिए मैं क्लिक करती हूँ ऑन दिस ऑप्शन मिरडीबी, ये एक अनदर ऑनलाइन टूल है जो कि प्रिडिक्शन टारगेट के लिए यूज किया जाता है। और अच्छा, यहाँ पे ये जो मिरडीबी एक सेपरेट डेटाबेस है, लेकिन इसका इस सॉफ्टवेयर का हमारे मिड बेस सॉफ्टवेयर में एक लिंक मौजूद है, हम वहाँ से इसे एक्सेस कर सकते हैं या हम इसे सेपरेट भी एक्सेस कर सकते हैं। यहाँ पे हमारे पास देयर आर 30 प्रिडिक्टेड टारगेट्स फॉर दिस माइक्रो आरएनए इन दिस डेटाबेस। और यहाँ पे हमारे पास एक टेबल है। पहले हम ये देखने से पहले कि व्हाट काइंड ऑफ द इंफॉर्मेशन वी कैन रिट्रीव फ्रॉम दिस डेटाबेस, हम ये देख लेते हैं कि यहाँ पे ये जो टेबल है ये हमें क्या इंफॉर्मेशन प्रोवाइड कर रहा है। टारगेट रैंकिंग, टारगेट स्कोर, मिरना नेम, उसका जीन सिंबल कि जिस जीन में इसका टारगेट मौजूद है वो जीन्स बता दी गई हैं, ठीक है। फिर आगे जीन डिस्क्रिप्शन है। इन तमाम में से जो टारगेट स्कोर है इज मच इम्पॉर्टेंट, क्योंकि जो टारगेट, और एक और पॉइंट ये है कि यहाँ पे जो टारगेट स्कोर है, अगर आप सारे जो टारगेट्स हैं इनमें देखें कि ये जो टारगेट स्कोर है ये 50 से लेके 98 तक इसकी रेंज है, नॉर्मली 100 तक होती है। तो इसमें यह है कि जितना ज़्यादा टारगेट जो स्कोर है वो हाई होगा, इट मीन्स यू हैव मोर कॉन्फिडेंस, स्टैटिस्टिकल कॉन्फिडेंस इन द प्रिडिक्शन रिजल्ट। जितना ज़्यादा स्कोर ज़्यादा होगा आप उसकी प्रिडिक्शन जो रिजल्ट है उसके ऊपर रिलाई कर सकते हैं। तो मैं यहाँ पे आई विल सेलेक्ट दिस वन। फर्दर इस तरह का एक हमारे पास पेज ओपन होता है जिसमें ये सॉफ्टवेयर हमें इंफॉर्मेशन प्रोवाइड करता है। किस तरह की इंफॉर्मेशन होती है? माइक्रो आरएनए नेम है, माइक्रो आरएनए सीक्वेंस, वी कैन रिट्रीव इट फ्रॉम दिस सॉफ्टवेयर एज वेल। प्रीवियस नेम अगर है तो वो बता देगा। टारगेट स्कोर क्या है, एनसीबीआई जीन आईडी क्या है, अगर हम यहाँ क्लिक करेंगे तो हम इस जीन की सारी इंफॉर्मेशन हमें एनसीबीआई से मिल जाएगी। जीन सिंबल क्या है, जीन डिस्क्रिप्शन, जेन बैंक एक्सेशन नंबर और कितनी लेंथ है वो बता देगा। लेकिन यहाँ पे जो इम्पोर्टेंट है वो है सीड लोकेशन। सीड लोकेशन क्या है या सीड सीक्वेंस क्या होता है? हम ये कहते हैं कि माइक्रो आरएनए हैज टू बाइंड with its target messenger RNA in order to down regulate it. लेकिन उसकी जो बाइंडिंग होती है, जो बाइंडिंग होती है एक्चुअली माइक्रो आरएनए और मैसेंजर आरएनए के दरमियान में, इट टेक्स प्लेस बिटवीन इन द सीड रीजन। तो ये सीड रीजन आइडेंटिफाई करना जरूरी है। आपको पता हो कि आपका ये जीन टारगेट है और इसमें ये वो लोकेशन है जहाँ पे हमारा माइक्रो आरएनए बाइंड करेगा और उस मैसेंजर आरएनए को या तो डिस्ट्रॉय करेगा, उसकी ट्रांसलेशन को इनहिबिट करेगा और जीन एक्सप्रेशन को डाउन रेगुलेट करेगा। तो वो इन्फॉर्मेशन हम यहाँ से ले सकते हैं और ये सॉफ्टवेयर हमें उसकी लोकेशन भी बताता है। जैसे यहाँ पे इस जीन के लिए 2 लोकेशन है, सीड लोकेशन 2 हैं, and starting from 303 position and the other one starting from 368 position. ठीक है, तो इस तरह की इंफॉर्मेशन आप सीक्वेंस भी रिट्रीव कर सकते हैं। यू कैन गेट सीड लोकेशन इंफॉर्मेशन फ्रॉम दिस सॉफ्टवेयर। ओके, फिर हमारे पास जो मैच्योर सीक्वेंस है 3p आर्म के लिए हम देखते हैं। इसमें एक्सेशन नंबर, प्रीवियस आईडी, सीक्वेंस, गेट सीक्वेंस में जाके इसके सीक्वेंस को रिट्रीव किया जा सकता है। डीप सीक्वेंसिंग इंफॉर्मेशन, जो एविडेंस हैं, एक्सपेरिमेंटल एविडेंस हैं वो, डेटाबेस लिंक्स, इसी तरीके से प्रिडिक्टेड टारगेट्स। अब यहाँ पे चूंकि मैंने 5p आर्म के लिए मिड डीबी जो कि एक सेपरेट डेटाबेस है उसको यूज़ करके दिखाया, तो यहाँ पे मैं इसको टारगेट स्कैन को ओपन करती हूँ। ये एक दूसरी सेपरेट जो है बायोलॉजिकल डेटाबेस है, ठीक है। और यहाँ पे ये जो हमारे पास टेबल ओपन हुआ है इसमें भी ये जीन्स हमें दिखा दी गई हैं कि ये टारगेट जीन्स हैं। इनके ट्रांसक्रिप्ट इन्फॉर्मेशन कैन बी ऑब्टेन फ्रॉम हेयर। जीन नेम क्या है, फिर नंबर ऑफ 3' सीक्वेंस टैग जिसको यूज किया गया था फॉर द कंस्ट्रक्शन ऑफ द 3' प्रोफाइलिंग, लिंक टू साइट्स इन यूटीआर। अगर हम इसके ऊपर क्लिक करते हैं साइट्स इन यूटीआर पर, तो हमें इस तरह की इंफॉर्मेशन, एक पेज हमारे सामने ओपन होता है जिसमें ये जो वाइट कलर का आप सीक्वेंस, वाइट जो हाईलाइटेड रीजन है वाइट, ये आप देख सकते हैं ये वो सीक्वेंस है और ये कंजर्व्ड सीक्वेंस है। दिस इज एवोल्यूशनरी कंजर्व सीक्वेंस अमंग वेरियस स्पीशीज, जैसे माउस है, रैट है, स्क्विरल है, रैबिट, ह्यूमन, चिम्प, रेसिस। तो इन तमाम स्पीशीज में ये जो सीक्वेंस है ये एवोल्यूशनरी कंजर्व्ड है और ये वो सीक्वेंस है जिसके साथ हमारा टारगेटेड माइक्रो आरएनए जाके बाइंड कर सकता है। फिर फर्दर अगर हम इसे स्क्रॉल डाउन करें तो हमारे पास 2 टेबल सामने आते हैं। एक टेबल है कंजर्व रीजन को दिखा रहा है और दूसरा जो है वो पुअरली कंजर्व रीजन को दिखा रहा है। तो यहाँ पे चूंकि 212-3p को इसलिए दिखाया गया है क्योंकि इसका क्लस्टर बनता है 132 का जो क्लस्टर है वो 212 के साथ बनता है, तो इसलिए यहाँ पे इस इंफॉर्मेशन को दिखाया गया है। और ये इनका कंजर्व रीजन है। यहाँ पे इनकी ये जो सीड सीक्वेंस है जिसके दरमियान में बाइंडिंग है वो शो की गई है। और साइट टाइप कौन सी है, अगर हम इस बारे में इंफॉर्मेशन लेना चाहते हैं तो हम इसके ऊपर क्लिक करेंगे और ये हमारे पास जो है वो इंफॉर्मेशन आ जाती है कि 8-mer साइट जो है वो क्या होती है। इसकी डेफिनेशन है कि इन एन एग्जैक्ट मैच टू पोजीशन 2 to 8 ऑफ द मैच्योर मिरना फॉलोड बाय एन ए। तो ये सीड सीक्वेंस है जो कि सीड सीक्वेंस एक्चुअली इट इज लोकेटेड एट द 5' एंड ऑफ द माइक्रो आरएनए। और फिर बाकी जब हमारे पास अगर इस तरह से कम्प्लीमेंट्री बाइंडिंग होती है तो हम इसे 8-mer साइट कहते हैं। और फर्दर इन्फॉर्मेशन जो कि 7-mer एमिड साइट है, इनकी भी इंफॉर्मेशन यहाँ से सॉफ्टवेयर से हम ले सकते हैं। और ये रेफरेंसेस इंफॉर्मेशन है हम वेरीफाई करने के लिए। और ये जो डेटाबेसेस यहाँ से हमने ओपन किए हैं इसे हम सेपरेटली भी ओपन कर सकते हैं। ये लिख नहीं रहा। ये ओपन नहीं हो रहा। ठीक है सर, थैंक यू। ओके, तो ये एक छोटा सा डेमोन्स्ट्रेशन था अबाउट द माइक्रो आरएनए सॉफ्टवेयर जिनको हम यूज़ कर सकते हैं जब हमने माइक्रो आरएनए स्टडी जो है वो प्लान करनी होती है। आप किस तरह से सीक्वेंस रिट्रीव कर सकते हैं। हम इसके अलावा इसमें जो है वो सीड लोकेशन की इंफॉर्मेशन हम देख सकते हैं और जीन टारगेट्स जो हैं उनको प्रेडिक्ट किया जा सकता है। तो ये सॉफ्टवेयर जो हैं इनके थ्रू हम ये सारा काम कर सकते हैं। सो थैंक यू सो मच।
Thank you very much for listening. Thank you so much.
Thank you so much, Dr. Mariam, for the nice presentation. Okay, so there could be two possibilities if you want to detect the miRNA from any source. Either we can detect the mature miRNA or we can go back to unmature form, which is stem and loop form. So which form is better for a diagnostic purpose or for to characterize it?
Actually, both these forms do exist, right? The first form, which is called the mature form, it is that one in which there is no stem loop, right? It is that sequence which is in FASTA format. And if you want to synthesize it artificially, like we design primers and then we send the commercial facility or sequence and then ask them to make those primers and send us back. This is how we use the FASTA format of the miRNA. But if you look at the functional consequences in the cell, inside of the cell, it is not existing in the linear form. It is in the form of a loop. So that software actually is depicting both type of formats for you. If you want to predict the functional consequences, where it is going and how it is going to attach with your DNA, you can have that form as well. And you can have the FASTA format as well if you want to synthesize it artificially.
What about the circulating microRNA? If we want to detect the circulating,
we are actually in our
which
project.
form it would be better?
In our project, actually, we are more of exploring the role of circulatory microRNA, and they are premature form. And once they reach to their destination, then they got their mature conformation, and then they got bind with their target.
Thank you so much.
Thank you. Thank you so much, everybody. Thank you.
Thank you so much, Dr. Mariam, for the wonderful presentation. And thank you, Ms. Ikra, for hands-on practice on the software. And thank you so much, all the participants. This brings ends to our fourth day of the training program. And see you tomorrow, the last day of the program. Thank you.